Hello, everybody. My name is Clarice Aiello. I'm faculty here at UCLA, and I'm chairing this very exciting, unusual invited symposium on quantum biology beyond photosynthesis, right? Because photosynthesis is maybe one of the poster child for this field of quantum biology, and I think that what we want to argue here is that there's enough evidence for the community, for physicists in general, to start thinking about other biological phenomena that might or might not include quantum mechanical rules at play. So I would like to spend 1 minute trying to share what I think one could mean by quantum biology, right? Because it's a very vague term. And the way that I came to think about it, and I propose that the community start thinking about it, is in two different branches, right? I think that there can be very good quantum-based biosensors, which may bring an advantage in probing biology at the nanoscale. One such sensor is, for instance, spins, the nitrogen vacancy sensor spins in diamond, which are very famous and have a very big hype right now. But also, there are potential bio-based quantum sensors, biological materials that might be using some laws of quantum mechanics in order to interact with their environment. That's why they're quantum sensors. And I think that the interest there is to, first of all, understand, either confirm or refute this, and understand how those bio-based quantum sensors might regulate physiology, right? And how one could, first of all, harness some of the lessons learned by nature over millions of years to inform the way we build better, say, quantum technologies, and even start to see whether we can control those quantum degrees of freedom to advance, say, therapeutics. This is a very short pitch for my lab. Please contact me if you have any questions. I am trained as a quantum engineer, and we're at the Quantum Biology Tech Lab, Qubit. We're interested in how quantum physics informs biology at the nanoscale. The group right now is joined by Dr. Lucas Ginsberg, who's a chemist, and Javier Carmona, who is a physicist and electrical engineer. And the idea is to start applying the same quantum sensing techniques that were developed to deal and control technological quantum sensors and start applying them to those putative living quantum sensors, right? And the mission of our lab is this one. Can quantum physics be established or refuted to account for physiologically relevant biosensing phenomena and be manipulated to technological and therapeutic advantage? So I'm looking forward to hearing from all the participants at some point, and I'm looking forward to a super cool invited symposium on quantum biology. So we will start with a presentation from Ron Naaman from the Weizmann Institute in Israel. Ron, can you hear me?
Yeah, I do hear you. Do you see my screen?
I don't.
You don't see my screen? Oh, I think you have to let me in somehow, apparently. Okay, 1 second. Now.
Yes.
Do you see my screen?
I do.
Okay, good. So, okay, can I start?
Yes.
Okay, so welcome everybody. It's pity we couldn't make it in live, but it's a good alternative. So I'm going to describe our work on electron spin and chirality. And we believe that this is the electron spin and chiral molecules are source for quantum effects in biology. I will try to briefly describe some of the work we did And if you have more questions or more information is required, don't hesitate either to ask me here or to send me emails. So commonly in chemistry and biology, one relates the chiral structure of molecule only to structure, not really to any electronic properties. And one relates spin typically to either ferromagnetic material or at least paramagnetic material. We usually assume that if we have closed shell system, the spin does not play any role. And what we found, and that's part of the message I would like to deliver, is that chiral molecules have unique electronic spintronic properties, and despite the fact that they are closed shells, namely typically singlet. So why it's important? Why these properties are important? I believe that these properties may answer several questions in biology. First of all, why are electron transfer in biosystem occur through proteins that are insulator and not through highly conductive molecules? Every student, if you will give him to design, him or her, to design electron transfer system, will pick something which looks like as close as possible to metal. However, nature picked protein, and the question is why? Can we understand it? Secondly, why chirality is conserved so persistently in biology all through evolution? And finally, how can nature be so enantioselective? We know that when chemists try to separate enantiomers, namely the two shapes of a chiral molecules, it's a really tedious work and it's very difficult. And actually reaching 100% is almost impossible. However, nature does it all the time. So the question is how nature does it and what is the secret that we don't know yet. So I will start in demonstrating a simple experiment in which we measured spin transport through chiral molecules. In this case, it's helicine. It's a chiral molecules that you see here. And the helicine doesn't have an asymmetric carbon, but it has a helical shape. And we use what contact magnetic AFM, namely it's a regular AFM that has a conducting tip, but the tip is also magnetic. We can use an alternative scheme in which we have conducting tip, and the substrate on which we put the molecules here is magnetic. And by switching the direction of the magnetic field, we are able to probe if there is a spin selectivity in the current that we measure. So here is the example of the result.
Sorry to interrupt, but the slide is still on the title page. We're not seeing the slides being changed.
What?
We only we don't see the slides being changed. Maybe if you could go to slideshow and.
I did it.
Yes. Now we see. Now we see. Yes.
Ah, that's the only way you see it. Okay, I will try to.
Thank you.
Okay, so this is the. Okay, so here it's the magnetic conducting AFM, and you realize here that we can measure using this magnetic conductive AFM. We can measure the topography of the surface. We can measure the current going through the molecule, and we can evaluate if the current is spin selective. So here are the results. Oh, it's not good. Something it's not good here. I don't understand it. Now you don't see it either? You don't see it? You see? Clarice?
Yes, we see the slides being changed. Yes.
You see now?
Those are the IV curves. Yes, we see.
Okay, good. So here you see the two IV curves for the two enantiomer, and clearly you see that for one enantiomer, the P, when the magnet is pointing down, you get higher current than when the magnet is up, and vice versa for the M enantiomer. And you realize also that the spin selectivity, it's quite high if we look at around minus 1.5 volt. You see that it's almost factor of 2 in terms of spin selectivity. Interestingly, if we look on this on the log scale, and do you see the log scale now? Do you see the log scale? Yeah, good. So if we put it on the log scale, you realize that the dependence of the current on the voltage is not linear. It's actually going as almost as a voltage in power of 2. Namely, we get a non-linear dependent of the current on the voltage. And if we measure the spin polarization as function of the length of the molecules, and here are the results for double-stranded DNA and for oligopeptide, alpha helix oligopeptides, we realize that the spin selectivity scales basically linearly with the length of the molecule. So that's interesting. So if I summarize this part of the experiment, it's important to appreciate that we can get very high spin selectivity of up to 85% in some cases. We see nonlinear conduction, and in the range of 10 of nanometer or more, we find linear dependent on the spin polarization on the length, and the effect is proportional, and that's something I didn't show, but I'm telling you, to the optical activity of the molecule. So what is the mechanism of the cis effect? Here is a simple diagram that shows that when electron is going through chiral potential, and any chiral potential is helical potential, electrostatic helical potential, and if you like, I can explain it, but any chiral potential is helical. So you realize that when the electron is going down this potential, for example, there is a force acting perpendicular to the velocity of the electron. Basically, it's the centripetal force. And in the rest frame of the electron, this force is exactly like a magnetic Lorentz force. So basically, the electron, it's in its center of mass, in its rest frame, feels as if there is magnetic field acting on it when it's going through chiral potential. And this magnetic field can act, of course, on the magnetic dipole of the electron, namely on the spin, and one spin become preferred and the other one is destabilized. Now, it's important to appreciate that the spin that is preferred depends on the handedness of the molecule and on the velocity. Namely, if electron is going in one direction, one spin is preferred, but with the same molecule, if it's going back, the opposite spin is preferred. And that's actually, you can also appreciate from time reversal symmetry. It's required that if one spin is going in one direction, the opposite spin will be on the opposite way. And this is an important thing to remember because it tells us that the spin is protected and that the spin is protected. So there is relation between the linear momentum and the spin, and the electron cannot change its direction of motion without flipping the spin, which is highly improbable effect. And I will elaborate on it in a second. However, this effect, what you see here, is giving us typically, when we calculate it, about 3 to 4% spin polarization. So how can we get up to 85% spin polarization? And for this, we have to take another step, and you realize here that when, let's assume that we have the chiral molecules between two electrodes. It's also true if we have it between donor and acceptor. And now when we apply the difference in the electrochemical potential between the two sides, charge in the molecule moves because the molecules become, as to charge polarized because of this difference in the electrochemical potential. When this electron in the molecule move, of course, one spin is preferred over the other, as I just realized and just mentioned, and although the difference is small, let's say 3%, now when the other electron wants to get into the system, it faces a 3% of an with one spin, and therefore there is a difference in energy for injecting one spin over the other, namely because of simple spin blockade effect. One spin can get easier into the system than the other, and we can estimate the difference in energy, and the difference in energy is basically the amount of spin polarization, let's say 3%, times the singlet-triplet energy gap, which is typically on the order of 1 electron volt. So in principle, we see immediately that we get a barrier difference of about 30 milli-electron volt for one spin to go through versus the other. So even a relatively small spin orbit coupling get enhanced due to the polarizability of the molecule and due to the nonlinear effect of the spin conduction. So basically, what this tells us that backscattering in chiral system is reduced. So that's already a hint that why it's so important for nature to have chiral system for electron transfer. In proteins, what is important is not the amount, the current that's going through the protein, because the number of electrons per second going through proteins in biology is relatively small, but what is important that every electron that starts to get in will get to the end, that electrons will not get stuck within the proteins, because then they will destroy the protein. So we realize that proteins are ideal system for electron transfer in biology because they provide a very good way to transfer electron with high reliability. Now I will switch to some other experiments in which we use whole device. Whole device is basically a device, classical device, where we have current flowing between source and drain. And if there is typically magnetic field acting on this surface perpendicular to the surface, then we get Hall potential, and the Hall potential is typically proportional to the magnetic field. It's a result basically of Lorentz force acting on the current. Now, we use this concept, but we do something different. We don't use magnetic field, external magnetic field, but we absorb on the surface chiral molecules. And now we use this setup in two configurations. In one of them, we have here chiral molecules on the whole device. Everything can be in physiological solution, like in biology. And we have an electrode here that is insulated from the solution. We apply voltage on the electrode. Because we have electrolyte solution, the field falls on the molecule, so the molecules get charged polarized. And we're asking ourselves, does this charge polarization in the molecules accompanied by spin polarization? Because if it does, then we will see Hall potential. In the other scheme, what we are doing, we are using this Hall device as the working electrode in electrochemical cell. It's a regular electrochemical cell. This is the working electrode. Here we have reference electrode and a counter electrode. And the idea is that we perform a regular electrochemical process, but simultaneously with measuring the current versus voltage, we also measure the Hall potential. So we get information about the extent of spin polarization in the current. And here, example of molecules, here we are getting to oligopeptides. And what we see is the following. Here it's the here's the electrochemical cell, and what you see here, it's the current versus voltage for both L and D oligopeptides. That's the regular structure that curves that people get in electrochemistry. But in addition, we get the Hall potential. So you see here on the right side that you see that for 1 enantiomer, we get the curve going this direction, in the other, the opposite direction. And that's because for L and D, we have different spin polarization. So that's a clear indication that we are able to measure spin polarization while measuring electron transfer through the oligopeptides. And here it's the other setup where we put voltage on a gate, and now using the gate, we apply different voltage and we look for the Hall potential. And you realize that the Hall potential here is increasing with the voltage applied, with the gate voltage applied. When we switch the other enantiomer, we get the opposite sign, again, because it's opposite spin. And here you see the structure, the Hall potential as a function of the gate voltage for L and for D enantiomers. But this tells us something very interesting. It tells us that in chiral molecules, charge polarization is accompanied by spin polarization. Now, this phenomenon is related to enantioselectivity. And what I mean by this, think about when two molecules approach each other, because of the induced dipole, there is a dipole, electric dipole formed. The electric dipole is accompanied by, of course, by charge separation, and the charge motion result in spin polarization. And what I just showed you is that the spin polarization, which spin is going to which pole, depends on the enantiomer. So for 1 enantiomer, we get the spin pointing up, and the other one, the spin pointing down. And that tells us something about enantioselectivity. You know that in chemists, biologists, assume typically that recognition of chiral molecules occur by lock and key model. And what we show here, this paper published by in PNAS about 2 years ago, that if two chiral molecules approach each other, and I make it very schematically here, you see that they get a charge polarized because of the induced dipole. Now the charge polarization is accompanied by spin polarization. And what you see that if the two molecules have the same chirality, then the interaction is through singlet state. However, if they have opposite chirality, the interaction is through triplet state. And therefore, what we claim here, that the chiral recognition is not only a special lock and key type effect, but there is an electronic term, which is basically the spin and exchange interaction that contribute also to the enantioselectivity, and that may explain the selectivity. And here you see calculation that show the different energy. The molecules here are simple molecules, the asymmetric carbons, and there is methyl group here, and we measure the interaction between the two methyl groups. So you realize between the two methyl groups, there is no special difference. There is no chirality in the methyl group, but the chirality is only in the back of the methyl group. And that's enough to induce different spin polarization that result in different in energy in the interaction if the two molecules are the same chirality or opposite chirality. And the difference is on the scale of several times kT. So it's a very significant effect. Now we will go back really into biology, and here I want to mention work that we did together with the group of Moel Nagar from USC. And basically the idea is to measure the spin polarization for electron transfer through two types of cytochrome proteins. One is more bulky. The left one here, it contains more alpha helices. The other one is less. And here you see the electrochemistry. So you realize that the one that is more bulky, the current is lower when we do the electrochemistry, while for the less crowded, the current is higher. But if we look for the spin selectivity, what you see here is the Hall signal. The Hall signal is larger for the more bulkier molecule. So it doesn't transfer charge so well, but the spin polarization is higher. And when we denaturate the protein, we lose completely the spin selectivity here. So the spin selectivity depends strongly on the secondary structure of the protein. And now I will give you a short introduction into another phenomenon. We have here, this work was done together with Dave Waldeck from Pittsburgh and Yossi Paltiel from the Hebrew University. And what you see here, that we have ferromagnet coated with very thin gold film. Very thin, I mean about 5 nanometer. We know that spin can transfer very easily through 5 nanometer gold. We don't lose much. And we absorb chiral molecule in it. And then we conduct what is called a contact potential difference measurement using a setup called Kelvin probe. The idea is the following. We have a counter electrode here that is, we vibrate this counter electrode against the surface with the molecule. And as a result of different contact potential difference between the two surfaces, the surface with the molecule and without the molecules, we get AC current due to this oscillation. We can apply counter voltage to zero the current, and that's voltage give us the contact potential difference between the two surfaces. And here came a big surprise. This part of the experiment was done using a contact potential Kelvin probe based on the AFM, and you see what happened here. For the D oligopeptide, we get for the magnet pointing, the south pointing up and south pointing down, we get two different potentials. We get the opposite potential for the L. And if it's not chiral, of course, there is no difference. So we understand here that what we have is that the current, when we apply this voltage due to the vibrating the surface, the ability of current to penetrate into the chiral molecules depends on the spin of the ferromagnet. So basically this picture is the following. Think about it, we have the molecule here. If the spin is aligned so that the spin on the ferromagnet is opposite, then this electron can penetrate in. But if it's parallel, it cannot penetrate in because of basically Hund's rule, Hund, if you like, spin blockade. So there is different for the ferromagnet being able to, ability to inject charge into the molecule, and it has to do with the spin polarization in the molecule. So basically we can think here about the spin as controlling the charge flow. It's the valve for the charge flow. And that's bring us to another type of studies that were done in collaboration with Gilad Aran from the Weizmann Institute. So we took a protein. This is the protein. We attached to the protein a dye molecule. And now we measure the adsorption of this protein on a ferromagnet surface coated with a very thin gold. And here what we see, if the magnet is pointing down, we get much less adsorption at short times than when the magnet is pointing up. However, if we wait long enough, then we lose the selectivity. So what it means that we have here kinetic effect. It's not thermodynamic effect, it's a different rate of adsorption. The rate of adsorption depends on the spin in the ferromagnet. And here you can see the selectivity. You see for 2 seconds, we have very high selectivity. And as we wait long enough, we lose the selectivity. However, there is selectivity. And now if we denaturate, then we basically use the selectivity almost altogether. So again, the ability or the spin selectivity of the protein depends on the secondary structure. But now we go one step further. Sorry, here. So what we do, we attach now antibody, that's the antibody, we attach the antibody to our ferromagnet. It's attached. Now we come with antigen that has a dye on it. And we monitor the adsorption of the antigen on top of the antibody. Now we can come with the antigen in two configurations. Either when the positive pole of the dipole of the antigen is pointing to the molecule, to the antibody, or the negative pole. So look what we get. What we see here, let's start with here when the negative pole is pointing towards the antibody, we see very high adsorption rate when the magnet is up, and much less so when magnet is down. With the positive pole, there is an opposite effect. It's not as pronounced as with the negative pole, but there is selectivity, and here it's summarized. So the question is, how can it be that we are able to control the binding here by changing the spin here on the button? And you have to realize that this antibody has a length of about 9 nanometers. So what's happening here? And the answer is related to what I showed you before. When the antigen tries to bind to the antibody, the binding is electrostatically, and it requires charge motion in the antibody. This charge motion depends on the polarizability of the antibody, namely how much charge the antibody can move towards the antigen. Now, the amount of charge depends on the spin here underneath, because if the ferromagnet is aligned in a proper way, charge from the ferromagnet can get into the antibody and effectively increase its polarizability. However, if it's the wrong spin, then charge cannot flow, which effectively reduces the polarizability of the antibody. And we see this opposite effect, of course, for the opposite direction of the dipole of the antigen. So we have here something very interesting by which we are able to control the spin polarizability, the polarizability of the protein by controlling the spin from the other side of the protein. So it's a new path for allosteric mechanism. You can imagine that if we have a molecule, instead of the ferromagnet, we have a chiral molecule that binds to the antibody from the other side, depending if this molecule is L or D, we will get different binding of the antigen. So that's a quantum effect which is based actually on the spin. So to summarize it, we observe spin selectivity in electron transfer process and protein interaction. The spin selectivity depends on the secondary structure. The spin can serve as a control for charge flow, affecting the electrostatic interaction of proteins. Now, answering you the question, I mentioned already that why proteins are good for electron transfer. Chirality is conserved, I believe, because it helps the electron transfer, it helps biorecognition, as I hinted, and something that I didn't touch, it helps in controlling redox reaction and avoiding byproducts. Why nature is so enantioselective? Basically, beside the shape, we have in addition to the shape, also electronic term in the interaction that helps the enantioselectivity. So these are the people, and thank you.
Thank you, Ron, for a very nice talk. Please, meeting attendants, post your questions on the chat box. Ron, I would have a short question, right?
Please.
So I would like to understand, as you know, more whether this effect is actually a spin valve or a spin filter that is spins with the wrong spin, electron with the wrong spins don't even go into the chiral potential, or can it also be understood as a sort of spin rotator? Are there models to say that if you have a certain
I have a very clear answer now. We just finished performing studies about it, and clearly there is no spin flipping within the chiral molecules. The filter is a passive filter, namely it doesn't allow one spin to go through, or it allows less than the other spin, but there is no spin flipping inside the molecule, 100%. And if you like, you can see it also in my presentation, because here you'll see, one second. You see the presentation?
Yes.
Okay, so you realize these are the two spins, the current coming from two spins, and you realize that they start at different energies. If there was spin flipping, we don't expect to get different threshold for each spin, because it doesn't matter which spin start, it will end the same, right? So the fact that we see clear difference in threshold for injection of each spin is indication that the chiral molecules are serving as passive filters and not as rotators. They cannot flip the spin.
Thank you, Ron. Phil Nelson is asking you to, when we upload the version of your talk, that you please include a slide with references.
Okay, good.
Thank you, Ron. I think we need to move on to Martin. Martin, can you take over? Martin, is Martin there?
He needs to unmute.
My video.
Unmute.
Okay. Video. Sorry. So let's see. Sharing. And do you see my slides?
Yes.
Okay, lovely. Okay, good. So let's get started. Oops, so this was already, okay. So here in this talk, I'd like to present a slightly more general view of certain aspects of quantum dynamics in biology. And in fact, I'd like to start with also pointing out perhaps what are the three main aspects that I see in this field broadly. And so these are, on the one hand, it's really our exploration of what role quantum dynamics and, as I will show later, noise actually plays in processes of life. So any kind of dynamics that happens at small scales in biological systems. So that's one aspect, and that certainly was the aspect that made me start to look into these things. But there's also, of course, the aspect that Clarice already mentioned, that it may also be that once we learn about these quantum effects and how perhaps to control them and enhance them and shape them, we can actually perhaps also use biological systems as different kinds of sensors with new capabilities, which take benefit from some of these quantum phenomena. And finally, another aspect that is important to me is that all these effects, it is very difficult to see them directly because most of our experimental technology looks at ensembles. And here I think new quantum sensors, new quantum technologies may actually be helpful to open new windows of observation into biological systems in general, and in particular into possible quantum dynamical effects. So for me, these are the three aspects of quantum biology that make it so worthwhile exploring it. And of course, there's a wide variety of phenomena that can play a role here. And so here, what I find also interesting about this is you see on the left upper corner, you see actually a classical machine, and it's cogwheels and stuff like that. And it is true that to understand the solid state physics of each of these materials, in principle, we may need quantum physics. But actually, quantum physics at that level is not important for the actual function of the device, for the dynamics, right? And this is because it's a very large system, which has a very small surface in relation to actually the volume of the materials. But what actually happens when we go to smaller scales, one thing that happens is that things become faster, smaller, and therefore have a chance to be more quantum mechanical, because noise does not have quite so much opportunity anymore to disrupt the system because the processes are relatively fast. And fast is always a question of speed of the process versus decoherence time in the system. So if you talk about spins, fast may mean nanoseconds. If you talk about excitons and charges, fast will typically mean picoseconds or femtosecond timescales. But of course, also when you become smaller, then the surface to volume ratio changes, and proteins are very much surface and not so much bulk. And therefore also in these processes, more noise or more interaction with the environment can seep in and actually have an influence on any quantum effects that you may see in the dynamics. And so this suggests that there may actually be a fruitful interplay of, on the one hand, coherent quantum dynamics, and on the other hand, the interaction, the noisy interaction with the environment, right? And if you look at the building blocks that you have typically you have proteins, you have molecules, you put them together in controlled arrangements. And of course, in reality, these arrangements are not static. They are in a body, a protein will shake around and it will move. And therefore, there is really a noise component to any dynamics that you may actually observe, right? And it's completely unavoidable. If we would like to avoid this, we would have to really go down to extremely low temperatures and to be only left with a quantum noise, which would be much, much smaller than what we have here. So it's unavoidable that we have noise, and it's likely that nature has actually made use of this to actually optimize its processes, and it may also have created a fruitful interplay between noise and the quantum dynamics in the system. Okay. So I'd like to touch upon very briefly, so let's say there are three main areas that have been popular. Photosynthesis, this has been in the old days very, very exciting, well, still very exciting, but there have been other effects that we have seen by Naaman, of course, but also here the question of olfaction and the question of magnetoreception. So I want to touch upon these 3 points a little bit, and I would like to draw the attention to the specific observation that each of these systems may exhibit quantum dynamics, but this will not be enough. They will only function well if there's also an interaction with the environment, so there is some noise seeping into the system. And I would like to bring out this point at the beginning. So in each of these settings, either demonstrated by experiments or predicted by theory, we'll see a performance, or let's say the behavior that you see on this slide, namely that when the system-environment interaction is very weak, then performance is not so good. If system environment is extremely strong, then performance is also not good because the system turns classical. And somewhere in the middle, at an intermediate system environment strength, so where the system is quantum dynamical, but the quantum dynamics is not completely destroyed and turned classical, we find sort of an optimal operating regime. Okay, so why could this be? So let's look at an electronic transport network here. And here we would have the challenge that we would, for example, like to transport an electron from A to B, or we would like to create a charge separation or something like that. So this could be part of a signaling chain, for example. And if we insert the electron at the top here, at the top level, and we would like to bring it down in energy to a bottom level, then of course, by the first law of thermodynamics, this will not typically work. We need some medium to get the energy into, so the energy difference. If we want to go from here to here, the energy difference has to be left somewhere. And this may be taken up by an environment. So this can be a vibrational environment, it can be a nuclear spin environment, it can be some form of environment. And if in this environment there are no typical frequencies that match this energy difference that we have here, then actually we will not see that there's almost no transport through the system. But then nature may actually start and tune this network in such a way that it adjusts the energy differences for the different electronic levels in such a manner that now the energy difference here is very well matched to dominant frequencies in the environment, so that these environmental degrees of freedom can actually take up this energy difference from the electronic system. And as a consequence, now you may have a transfer of the charge from one side to the next, and therefore eventually through the entire system. So there can be a way in which we tune electronic systems to match vibrational or other external degrees of freedom in such a manner that energy may be exchanged. And this will only work if, on the one hand, there's enough coherence in the system to allow for this tuning of these collective energy levels. But of course, there has to be a sufficiently strong interaction with the environment to be able to transfer these excitations of the energy from the electronic system into the environment. And this sort of suggests that indeed there is an optimum at an intermediate system environment interaction strength. And this is just merely one example. There's a whole range of effects. Each one of them will benefit from some quantum coherence and from some noise, but only when the two are well matched in strength in the system. Okay, so here you can actually play a little more game, and this will drive us to the theme of actual sensors based on biology. And here we would see, let me assume that here in the environment, I actually add a degree of freedom that is very, very well defined. So it's very narrow, spectrally very narrow. Okay? So that means it might be a vibration that has a very long coherence time, or a nuclear spin or an electron spin that has a very long coherence time. And if this happens, then actually we will not merely bring excitation into the environment, but actually it can also come back. And so we will actually see sort of coherent oscillations of the electronic degrees of freedom and this external environmental degree of freedom. And these kind of oscillations, it should be possible to see if we want to show that indeed there is this kind of interaction between quantum dynamics and vibrational motion. Now, this is very close to photosynthesis, but actually it's organic photovoltaics, where actually we looked experimentally into this question of indeed charge transfer, that means electron transport in the end, can actually be facilitated by, in this case, vibrational motion in the environment. And indeed, what we see experimentally is that the presence or absence of very specific frequencies in the environment make a big difference to the quantum dynamics of the electronic system. And what you will find actually is that here we have a dynamics where we see actually the electronic dynamics of the system, and more specifically we see the charge separation process. So when we have low values here, the charges are still together. Then the larger the value here, the farther they are separated, and so the more the electron has actually propagated in the system away from the hole. And if we take the full model, which we derive from experiment here, we have the electronic dynamics, we have specific vibrational modes, we see that actually the charge separation, the black line here, is very rapid. So very rapidly you go to 75%, you have really removed the electron very rapidly from the site where it was created. But now as a theoretician, what we can do is we can leave all the model parameter the same. With one exception, we change the environment. That means in this case, we remove this one vibrational mode. And then we see a dynamics of this type, where the black line rises much, much more slowly. And what this really means is that this environmental degree of freedom here actually helps the propagation of the electron through the system and helps the charge separation. This is not a direct experimental proof, of course, but it is the best, let's say, that we can see with experimental techniques so far, that really environmental degrees of freedom can help electronic dynamics here. But now I would like to go a little bit further and look again at this whole process, but turn it around and turn it into a sensing device. And this is an idea that was suggested initially by Luca Turin, but actually it turns out it's also an effect that is used in condensed matter physics quite regularly to measure vibrational spectra. And so now let us imagine we have a situation of the following type. So here's a very abstracted view of a membrane receptor with a binding pocket here. And it has an electron source and an electron acceptor. And if there is an energy difference between these two, then the electron cannot simply jump from the source to the drain, because it has to get rid of the energy difference somehow. And if there's no vibrational degree of freedom or any other degree of freedom in the environment that can take up this energy difference, there will be no energy, there will be no electron current. Okay, so now imagine a molecule comes along and sits in the binding pocket of this membrane protein. And as a consequence now, we've brought close to the donor and acceptor here, we have brought vibrational degrees of freedom that are sitting in the molecule. If one of these vibrational degrees of freedom, one quantum of this vibrational degree of freedom has an energy that matches the energy difference between donor and acceptor here, then now what can happen is that the vibrational degree will be excited and the electron will move from donor to acceptor, and therefore creating a signal in the protein that tells us, aha, there's something in the environment, some molecule that actually allows us to make this electron transfer. So this can serve as a detector for the presence or absence of a particular vibrational degree of freedom, and therefore of particular types of molecules. Of course, here what we would not like to have is that this electronic degree of freedom could get this vibrational could give back its energy to the electronic system. So therefore we will have to have some dissipation in this vibration so that it gets rid of its phonon sufficiently quickly not to have a back transfer again up from the acceptor to the donor. Alternatively, in the acceptor, the electron has to be moved away sufficiently fast so that this back transfer becomes impossible. So in a sense, this transport process that we have seen before here, it is really used as the presence of transport is used as a detector for a molecule. So in this sense, this is a biodetector, and it depends on some quantum mechanical features of the interchange of interaction between the electronic degrees of freedom and here the vibrational degrees of freedom. So now the next step is magnetoreception, and I will be very brief here because Thorsten Ritz will of course give a much more broader view of this. But I would like to bring out here yet again that this is a phenomenon that really does not only depend on quantum dynamics, but actually also on noise. And what is the basic feature here? Also, the basic idea is that we have a molecule here. A photon will come and excite an electron pair from the ground state and create a radical pair. And because it emerges from the ground state, it's initially in a singlet state, and so the initial state of this radical pair would be a singlet state. And now a singlet state itself has no angular momentum, therefore it's completely rotationally invariant, and this alone cannot share any sort of, let's say, magnetic field sensitivity to, let's say, an externally applied field. It has the same energy no matter what the orientation of this magnetic field might be. However, we have to be realized that, of course, these two electrons, they have been excited, they are moving around, and they will see local environments that are not identical. And they will actually start to interact with nuclear spins in the molecular substrate on which they are, or the protein substrate on which they are living. And as a consequence, because they see local interactions, they will actually start to see some local dephasing. And what this does is, it actually turns a singlet state, or it can turn a singlet state into a spin triplet state. So now we have another ingredient, namely we have the singlet to triplet rotations that are generated by the interaction with nuclear spins in the environment. On the other hand, we will have a quantum mechanical dipolar interaction between those donor acceptor, between these two electrons in the radical pair, or rather between their spins. And this quantum mechanical interaction will depend on the relative orientation of an externally applied magnetic field and the vector joining the two electrons in this radical pair. So it's really now again this convolution, or the combination of this quantum dynamics in the electronic spin system and its interaction with the environment, namely here the nuclear spin. If there was no nuclear spin, no environment, the electrons would be initiated in a singlet state. They would simply not be able to show a different type of response depending on the applied magnetic field. And then the last step in this procedure is, if they are falling back into the ground state, they can do this easily when they're in the singlet state, but not so easily in the triplet state. And so this will affect subsequent molecular processes. And again, I think Thorsten Ritz will be more specific about these points, but here the main message is, again, we have quantum dynamics and we have an interaction with an environment that will try and make the system more classical. And if these two effects are well balanced, we will get an optimal function for the dynamics of this particular sensor, which would in this case be a magnetic field sensor. Right, so these are sort of phenomena, and some of those, in the photosynthesis arena, let's say there are things can be investigated rather well with laser spectroscopy, but we have a challenge here that we always look at ensembles. In the other two effects that I've shown, olfaction and in fact here magnetoreception, it's actually very difficult to even use experimental technology to make direct measurements, because these will not be susceptible to, not so easily interrogated by light, because we are talking about spin effects here. So really, it is very important that we develop new sensing technologies that can either go away from the ensemble and look at individual realizations of a biological system and look at the processes that are taking place there, or actually develop sensors that are, for example, magnetically sensitive, that we can bring close to, for example, an olfactory receptor, or here the proteins that are held to sustain the magnetoreception, in order to make rather direct measurements of the dynamics of these systems, so that we have individual systems. We are not losing coherent phenomena that by dephasing over a large ensemble. And therefore, there's really a case for trying to develop new also technologies for better measurement to test for quantum biological effects. And there's a good tradition here in that physics has often learned to harness new physical laws or physical phenomena and turn them into devices that are used to then examine biological dynamics. So the first example of that is perhaps the microscope, where actually 400 years ago, physicists have learned to use lenses to actually manipulate light. And that allowed actually a Dutch scientist, van Leeuwenhoek, to discover the presence of bacteria, which was completely unknown before. And of course, nowadays we have super resolution microscopy and all these things, and we can really look deep into biological systems. But it's still, the spatial resolution is not yet very, not good enough to really look at, let's say, quantum phenomena on single systems. But of course, we have also X-rays, we have nuclear magnetic resonance, and all sorts of other phenomena that have allowed us to look deeper and deeper into biological systems. And in the spirit of this progress, so what is happening now, and it's also work that I am kind of involved in, is that we are trying now to harness actually the dynamics of individual electron spins in a solid state matrix, typically in diamond actually, control them so well and bring them very, very close to our target materials to use those electrons as magnetic field sensors, and which are capable actually of seeing individual nuclear and electron spins. And if it becomes possible to move these systems close to a biological system, then it may well be conceivable that we can actually examine some of these effects in olfaction and magnetoreception. And just to give you a brief idea of how these sensors may work, so here is a diamond lattice, and you can see here a very small lattice defect where a carbon atom has been replaced by a nitrogen atom, and a vacancy has been created in the neighboring sites. And you have dangling electron bonds here, and they create actually an effective electron system, which is a spin-1 system. And it has an energy level structure of this type here. So you have an m equals 0 state, and then separated from it, you have an m equals plus and minus 1 state. There's an energy separation here. And these energy levels here, they will shift when there is a magnetic field applied to this defect center. And this magnetic field can come from a single electron that is maybe only 10 nanometers or so away from this sensor. So if we create these defect centers to within a few nanometers of a diamond surface, and then place an electron spin or a nuclear spin for that matter close to this surface, then we will actually be able to see this electron spin. So it's to see the magnetic field that it creates. And in this way, we have a magnetometer that can actually sense individual electron or nuclear spins and their dynamics. And this would, of course, hopefully open the possibility to do experiments of the type where we have here, cryptochrome for magnetoreception. We would bring a very small nanoscale diamond very close to this system, and then use it to actually study the electronic dynamics in this radical pair. For example, how quickly does it recombine, what is the spin dynamics. And when you go through the parameters, it's a challenging experiment, but it's not completely out of the question to do something of that type. You can also think that you can bring such a diamond close to a membrane receptor and then try and observe the arrival of a molecule and maybe structural changes and electron transport in such a membrane receptor. Again, very challenging. One has to develop technology, chemistry in particular, to make selective binding and to bring the diamond here close enough and all these things. Again, challenging experiments, but not out of the question that they actually become possible. And certainly first steps in our group together with colleagues we have taken, where we actually inserted diamonds into a cell, and we used, started to learn to control the electron spin degree of freedom of this one defect center in these nanoscale diamonds to try and make it robust to environmental noise, while at the same time still make it sensitive to actual signals from the environment. There's a whole lot of techniques that have been learned in the last 10 years, and they have been typically applied to bulk diamond, where the color center was inserted there, and they were very well controlled, and it was very difficult to see how one could maybe do these same techniques in nanodiamonds inside of a cell. And this is a demonstration that indeed we can actually do so, and we can in principle achieve quite significant magnetic field sensitivities that may allow us actually to monitor the spin environment inside a cell using such diamonds. And so with that, I would like to close with this view. So here's some literature that I have been using. So, so this slide is a little bit messed up for some reason. Interesting, yeah. So you see in the background the European Science Foundation report that has brought out the various aspects of quantum biology, and also actually challenges that at this time at least we have been perceiving in the field. And so this may make interesting read, and it's available online on this link here. And the other setting is, so the story that I've been telling you about coherence and Kurian dynamics and noise, you can read a slightly older version of it. You can read in this article here in Contemporary Physics, which is an overview of the various phenomena where noise and coherent dynamics shows an interplay. And when you want to learn about these quantum sensors that we are interested in, this is a review where we look at these quantum devices, namely color centers and diamond, and their potential applications in biological systems. And so with that, I come to the end. Here I have also a slight advertisement slide for our new institute. So almost 10 years ago, I had the plan of creating a center which brings together physics, quantum physics, and biological sciences and medical sciences. And well, after some work, this is a schematic drawing. Here's the real thing. So this is the photo of the center, which has now opened. And it hosts theoretical quantum physics, experimental quantum physics, diamond sensors, in fact, medicine and chemistry, all working towards the goal of really trying to build new quantum technologies for sensing in biological systems, hopefully to study quantum effects in biological systems, and also to do theoretical work towards, quantum effects in biological systems. And then hopefully translating this also into medical applications with the help of the medical teams in our building. And this is the team and the funders, and with that I would like to close. Thank you very much.
Thank you, Martin, for this very broad, interesting talk. There is one question from the audience, and I am going to ask you to answer one question. So let's start with the one question that is, I'm going to make you address the elephant in the room, the question number one of all physicists. What about coherence types? Can you?
What about coherence?
Yes, coherence types in general.
Types.
Yes,
Okay.
that is possibly the question number one when I interact with physicists.
Indeed. So
And,
let me go way back here. I would say, so here, it is clear, of course, and it's a very valid question, how can it be that we have coherence in such a system when they're warm, wet, and noisy and not isolated from the environment? And the coherence times that we have to, that one can see depend very much on the system that you're using and its strength of interaction with the environment. So if you talk about excitons and charges, like we do, for example, in photosynthesis, then perhaps the coherence times are in the range of, let's say, between picoseconds and femtoseconds, rather on the lower end of this scale, because charges and excitons interact relatively strongly with their environments. But this is counteracted by the facts that these dynamics are also very fast. So the interactions are strong, the dynamics is fast, and therefore quantum mechanics can still play a role in these effects, because although the coherence times are short, the relevant timescales of the processes that are taking place are equally short. For electron spins, you can expect coherence times perhaps closer to a nanosecond scale. And for nuclear spins, perhaps microsecond scales. And again, here, of course, then the question is, are the processes of the spin-spin interactions fast enough to actually allow us to see some coherent electron spin dynamics, for example? And when you look at this and you look at sort of typical separations that you would create for example, in a radical pair, then you will see that the dipolar interaction between these electron spins can actually also be in the hundreds of megahertz regime, or even higher, depending on precise distances. And so therefore, coherencies again can be long enough for the specific processes here to actually play a role. Yeah? And so it's always an interplay of coherence times, that means interaction with the environment, and the speed at which the dynamics is taking place. And typically for all of these phenomena, you can find sweet spots where system-environment interaction is there, but it's not yet so strong that it completely suppresses the coherent dynamics in the system.
thank you, Martin. And now there's a question from the audience about the nanodiamonds. How do you envision functionalizing the nanodiamonds, the nanodiamond surface? Example, could you please give some examples?
Okay, so this is of course, this is a challenging question, but a lot of progress has been made. So what you can do so there are different strategies. One strategy is that actually really you treat first you clean the diamond surface, let's say with acid treatment. Then you provide COOH bonds on the surface. And then by click chemistry, you can actually attach to those bonds, you can actually attach further groups, chemical groups, and therefore build up the surface. So this is possible. Another possibility is that you actually wrap these nanodiamonds in proteins. So for example, human serum albumin is a possibility where you can actually wrap the diamonds in this protein, and this protein itself then will have attached labels that are like barcodes that can then selectively bind to to other target proteins that we want to bind to. So in principle, steps of this nature have been taken in chemistry, right? So Tanja Weil, for example, that is also part of our center here, actually has developed chemistry to create this kind of functionalization on diamond surfaces. The next step is really, of course, to show that we can really make the right sort of label to create really site-specific bindings of these diamonds. And a challenge at the present is that these diamonds together with, let's say, the coating around them, are relatively large still. So I think we still have to make them a bit smaller so as to be able to actually really create the specific sites on proteins. So this is a challenge, it's clear. That's why we need the chemistry activities in our center also to actually really realize this, surface functionalization and specific binding. But there are clear routes on how to do this. And you will find some of this information also in this review article that I mentioned at the end of my talk concerning quantum devices and biological systems.
Thank you, Martin. Thank you very much. And I think we need to give the hostship to Philip Kurian. Mm-hmm. Philip, are you there?
Can you hear me, Clarice?
Yes. Yes, we can.
Okay, great. Let me share my screen. Can you see it yet?
No, I think I probably have to stop mine first. So, okay. Yeah.
Okay.
Yeah, and it's recording still. Okay.
Okay.
I don't see Philip's screen.
Oh, it's a moment ago it was here.
Do I have to click the share screen, Clarice, or do you?
You have to click share screen.
All right. Is it under advanced sharing options?
It should be at the bottom.
Okay, I got it. Yeah, here we go. Okay,
awesome.
Let me know. Can you see it?
Yep, perfect.
Yes.
Okay, excellent. Well, thanks, Clarice, again for spearheading this effort to create this virtual session in this space. I think Martin and Ron have given a really nice introduction to some of the major themes, which frees up some time for me to go into some work of the quantum biology lab at Howard, where I serve as the director. I'm going to present across a number of different research foci. Hold on. The co-host has asked you to start my video. Okay. So I'm going to talk about sort of 5 general topical areas revolving around some horizons of quantum biology that have been touched on in the previous 2 talks, but have also maybe some that have not been. And so particularly I'm going to start with this look at plasmon-like allosteric enzymes, which mechanical allosteric has been discussed quite a bit, even in the biological and biochemical literature. But how could these plasmon-like waves be implicated in the synchronization of protein and enzymatic processes? And then I'm going to talk a little bit about work on superradiance in highly symmetric biopolymers, and work with collaborators Luca Cilardo in Benemérita Universidad Autónoma de Puebla, and Travis Craddock at Nova Southeastern University. And then we're going to move into some discussion of how in the noisy electrodynamic milieu that is the cellular environment, how do we conceive of proteins and their cognate partners finding each other at the right place at just the right moment, at just the right sequence. And then describing that in both a sort of photon to phonon transduction process, in that we can both see the optically induced mechanical restructuring of proteins, and how could that be communicated in a complex aqueous environment. And then I'm going to conclude at looking at how these electronic effects that we started describing at the very fast quantum level, and nuclear quantum effects that describe phonon modes, how they might interplay. So the work that we're doing in plasmonic allostery is in partnership with the group at the University of Luxembourg, led by Alexander Katchenko, and also with Martin Storr. But it began with looking at a very particular enzyme system that has some unusual properties. In 2016, we published this paper in Theoretical Biology, asking the question of how this EcoR1 enzyme complex is able to synchronize the breaking of 2 DNA strands at significant distance, in excess of 20 angstroms. And it's able to do this with an amazing efficiency, and it's able to do this in a way that's somewhat simultaneous. And simultaneity, of course, being defined by fast relative to thermal equilibration. And so the question is, so how does this enzyme function? The biochemists have studied it for nearly 60 years now, because these enzymes because they target a very specific sequence of DNA. They were used in the revolution in genetic sequencing and engineering that precipitated the Human Genome Project at the beginning of the 21st century. So they recognize that they're able to solve two problems very rapidly. They can, one, identify this target DNA sequence of 6 base pairs in the human, in a bacterial cell, in millions of base pairs. They're able to do this very fast, faster than what would be predicted from Brownian motion. And then once they bind their target sequence, they're able to synchronize that catalysis of 2 phosphodiester bonds, initiating the double strand break. And then after that, this water floods into the enzyme complex, and then the enzyme dissociates from the DNA, and it is the DNA strands are separated from the thermal motions of water molecules. But if we think about this protected state, you can see here in the middle of the figure here, where this DNA, the enzyme complex, is able to scan the DNA sequence in a sort of non-specific configuration. And then when it binds, it clamps down on the DNA sequence, excluding more than 100 water molecules from the surface of the DNA complex. Is it possible that quantum correlations between the DNA base pairs, what biochemists have called substrate-assisted catalysis, but never identifying a quantum feature that corresponds to such catalysis, is it possible that this kind of behavior could be responsible for the synchronization between the catalytic sites? And so this back of the envelope calculation, which we did, showed if we treat these base pairs as quantum harmonic oscillators and we couple them linearly via their polarizabilities and a dipole-dipole potential, we can get an agreement within about 0.5% between the 0-point modes of that coupled system and the energy required to break these 2 phosphodiester bonds. What's interesting about these enzymes is that they don't require an external chemical energy source to move over the enzyme transition state, which is why that some of these models have been proposed for looking at how the substrate could be used by the enzyme to overcome that barrier. So as I mentioned, a more refined model of these electronic quantum fluctuations has been developed by the group of Alexander Tkatchenko in the framework of extended van der Waals interactions, or so-called many-body dispersion modes. So you can see here the Hamiltonian for such modes, which represent a coupled system of quantum Drude oscillators that describe these fluctuations, how they can couple at long range. And a key point is that these parameters that are derived, so you have these this interaction tensor that describes the interaction between your coupled system of n quantum harmonic oscillators. These polarizabilities are derived from advanced electronic structure calculations using density functional theory tight binding. And so the key point with these many-body dispersion modes is that they cannot be written in a pairwise fashion. And in fact, if you open any biology textbook and you look at pairwise van der Waals interactions and how they're treated between individual sites in your complex system, the standard sort of dogma around van der Waals in biology is that they have no relevance beyond, let's say, 2 or 3 nanometers. But in highly symmetric systems, or in systems that are coupled to an aqueous environment, we are now seeing, particularly in this paper, that these systems can actually exhibit couplings that extend far beyond that range. And so in this paper, what we can show is that if you look at the near range aqueous environment around some small proteins like chignolin, which has a very specific protein structure, or some other beta sheet or alpha helix structures. You can see that if you use the pairwise van der Waals approach, you can only see effects out to about 2 or 3 water layers. In the red here, you can see those pairwise van der Waals. But then if you include the effects of the many-body dispersion, where at this frequency range, the dielectric constant of water is about 2, so almost metallic, you not only see this extended effect that extends to about 30 angstroms, which is also a product of the box size used and the protein configuration chosen, but we can see these plasmon-like correlations that are occurring within the protein complex and far outside in the aqueous environment. So this is a coupled eigenmode, a sort of plasmon coupling across both within the protein core and outside in the aqueous water molecules outside. And this has an important contribution to protein folding, with up to a 20% jump in the solvation energy as determined from MBD. So what are these, how can we visualize these modes and think of a way to look at the information content in these modes and how it might be communicated in a biological system? These are very fast modes, but yet there is, and this is a current active area of research, is how this petahertz scale modes could couple to the phonon modes, which are 3 to 4 orders of magnitude slower. And so there's a very interesting way of approaching these forces that these plasmon modes exert on the nuclei. And these nuclei can then couple to experiments that we currently have available. So one thing we wanted to do was to look at the collectivity of these modes, because in biology, I mean, these MBD modes are truly delocalized across the entire complex of your system. So this is for a system with n atoms, which this system has about 9,000. You have 27,000 modes. And so it's a big data problem to look through that. And so my postdoc, Matteo Gori, has done an incredible job of looking at how the Shannon-like entropy of these modes can be a tool for analyzing both the centrality of eigenmodes and their network information content. And so if you can see here, this is one example of one of these modes, and you can see these magnesium ions. These are magnesium cations that couple into the system in a very symmetric way. And at non-optimal biological concentrations, where the symmetry of this complex is disturbed, you actually see a non-efficient process where the enzyme is only able to cut a single portion of the DNA at a time, and then it's slowed down by the process of having to unbind from the DNA complex, go through the process of scanning again, rebinding, and then cutting again. So there's a sort of a dual but much slower process that occurs in non-optimal conditions, which brings up this whole point about the robustness of biology across multiple scales, which I'll touch on a little bit more later. So these eigenmodes, to each of these eigenmodes, which have 3n components, we can associate a probability to each of the atomic sites, which is the sum of the squares of these normalized eigencomponents for the given a single atomic site. And those probabilities can then be associated to a Shannon entropy. And what was curious that we noticed at first was this correlation between the density of states in black and this normalized Shannon entropy that we see, these sort of 3 distinct bands, which could roughly correspond to the polarizabilities of carbon, hydrogen, and oxygen. But of course, in the density of states, you have this coupling that occurs across the structure. So we define this set of mathematical tools to look at this space of eigenmodes, where we have this participation probability that I just described as the sum of the squares of the normalized eigencomponents for each atomic site, and then this degree of collectivity, so that when you have your entropy equal to 0, you have all of or the vast majority of the eigenmode situated on a single atomic site. But then when your entropy is 1, you have a very noisy distributed participation probability. And so here's what that looks like. So in the case of a high entropy mode, you can see that it may not have a functional purpose in biology for allostery, but there may be some other purpose for how these plasmonic fluctuations may be influencing the biological system. But you can see here, this is the graph of the individual probabilities of those sites on individual residues, where here you have the DNA bases, and then you have some key residues in the catalytic site. And you can see that the highest probability here is on the adenine that is actually involved in the process of the DNA double strand break. But then in this medium entropy mode, which we find that that medium entropy case is the case where you have sufficient number of residues that are involved in your MBD mode, but then there's some spatial separation also, in that it's not just on a single atom or a single residue, but that it's somehow optimized for complexity of the system. And here again, you see the high probability situated on a single DNA base. So what we've done is to look at this 27,000 space of 27,000 modes for this system, and trying to define an algorithm for looking at which modes might be most interesting from the perspective of allostery. And so you can see in this, there's almost a chirality to some of these modes. And this mode is highly situated along the DNA complex and involving those phosphodiester bonds along the sugar phosphate backbone. But what we can do is we can choose, depending upon the system, what kind of residues we're looking at, whether we want to look at the DNA bases, whether we want to look at the catalytic residues that are involved in these magnesium pockets. And so we can pick those relevant sites and then compute these probabilities to see, let's say, what is the centrality of a given node along these complexes. And so here was that algorithm that was run. And so I don't know if you can see if I zoom in here, but again, you see this chirality of this given mode. But then also here, what we did was plot those probabilities on the two catalytic sites where the phosphodiester bond is cut, as well as for those catalytic residues. And then we constructed an adjacency matrix from all of the pairwise probabilities products, and then computed a sort of centrality measure by looking at the eigenvector components for the largest eigenvalue of that adjacency matrix. And you can see here we have sort of a MBD forensic for each of the eigenmodes, where here are the two catalytic sites with the magnesium ions, and we could just as easily have chosen a different set of residues to define what our probability or centrality is. But here you can see very clearly that those two catalytic sites become the central nodes in our network for the allosteric recognition and catalytic process. And so this eigenmode here, which is ranked 7th in energy value. And so this energy value here seems very large in terms of electron volts, but that's because it includes the electronic structure energy. If you were to subtract off the system, the expectation value of the uncoupled system of oscillators, we would then have energies closer to between 0.1 and 1 eV. So I'm going to switch gears to the work that we've done with Luca Cilardo and Travis Craddock. Clarice, can you just give me a sense of how much time I have?
You have about 10 minutes.
Okay, great. So this is a system in the cytoskeleton called the microtubule, and the cytoskeleton forms the structural integrity of all cells, and it's particularly important in neurons. But what you can see here is that in these structures, you have a very unique spiral cylindrical symmetry of this protein molecule tubulin that forms into these 13 protofilaments that then forms this cylinder. And these, within these protofilaments and within these microtubules, you have a network of tryptophan chromophores, which absorb in the ultraviolet. And then they have a very strong transition dipole moments of about 6 dBi. And those are very comparable to the transition dipole moments of chlorophyll, which you can see in photosynthetic nanotubes. And so we wanted to look at how do these tryptophan chromophore networks in microtubules, how might they couple together? So we use this non-Hermitian model, which was developed by Shaul Milkamel and has been used by others widely in the quantum optics community, to look at the case of when your photoexcitation is much smaller than your system size. And so these microtubules can be tens of microns long. In our case, we look at one that is 1 micron in length. And we can show in this formalism that you have couplings that have 1 over r, 1 over r squared, 1 over r cubed terms, and these reduce to the simple dipole-dipole couplings when your system size is very small. So what are our results? This big gamma here is the eigenvalue of that diagonalized matrix that I showed on the previous slide. And these small gammas here are the single tryptophan decay width. So what we see is that with increasing symmetry, as you add the number of spirals, your lowest excitonic state increases in its superradiant strength, such that when you get to 100 spirals, about 1 micron, that's 10,000 tryptophan chromophores, you're at 600 times the single dipole decay width. And what we were curious about was, is this a product of native symmetry in the microtubule because we use the actual orientations of the tryptophan chromophores, or is it something that could be reproduced? And so we fully randomized all of the dipoles, but kept their positions, but randomized their orientations, and we wash out completely the superradiance. The other case is where we take a single spiral, and then we randomize the orientations on that single spiral, but then we repeat it so that we create some structure, but it's not the native structure. And you can see we reproduce some superradiant effect, but not to the extent of 100 spirals. And so this is the wave function of our coupled system projected onto the chromophore basis. And what we notice here is on the top, you see this is the superradiant state plotted on the microtubule of 1 micron in length. And on the bottom, and this is in longitudinal and cross section. And then in the bottom, we have the subradiant state, or the most subradiant state at the middle energy eigenvalue. And what we notice is that these superradiant states, which exist on the few picosecond time scale, these tend to localize on the surface of the microtubule, whereas these subradiant states seem to locate themselves on the inner side, the lumen. And so this suggests 2 divergent time scales at which we could possibly measure these effects, because whenever you have a superradiant state, you have a complementary subradiant state. And so these superradiant states can be measured with ultra-fast spectroscopy, and we're working with Majed Chergui at EPFL to measure the superradiant redshift signature of this state. And then these millisecond subradiant states could be measured even at the level of seeing fluorescent spectroscopy or other types of slower spectroscopy. So why might this be relevant in biology? In 2017, we published this paper looking at how reactive oxygen species that are emitted by mitochondria actually emit photons. And this has been well known from 300 to 350 nanometers. We can detect these photon emissions that have been emitted from triplet carbonyls as well as singlet oxygen radicals. And is it possible, we haven't been able to detect at 280 nanometers, but is it possible that these emissions, these ultraviolet emissions, could be used because these mitochondria co-locate with microtubules in neuronal axons? And could this be a potential for signaling that's stimulated by reactive oxygen species? So I'm going to breeze through this a little bit, but to look at long-range signaling in biomolecular systems, we need intelligent, robust adaptation to environmental demands. And what does robust mean? I mean, biology is both slow and fast. It has, optimized over 12 to 18 orders of magnitude. And so we have couplings between every scale. And so what are the correlations between things happening at the femtoscale to things happening at the pico to nanoscale? That's a big question. So as I mentioned before, we have this highly coordinated dynamics in space and time. This is just a single pathway in the complex biomolecular environment. But we have this recruitment of cognate partners at just the right moment in just the right sequence. So what are the physical interactions at play in these biomolecular dynamics? So work with Marco Pettini's group at Aix-Marseille University in France has challenged this conventional idea that only short distance or at short range, we only have these interactions, but then at long range, there's only sort of mesoscale kind of coarse grain descriptions of hydrodynamic interactions and stochastic forces. So what about long range electrodynamic interactions? So in the 1960s, Herbert Froelich proposed this idea that out of equilibrium systems like biology with a rate injected energy can produce these giant dipole oscillations in the terahertz regime. And these terahertz modes are not screened by Debye screening due to the electrostatic environment, and they have a lower dielectric constant value at these speeds. And Marco Pettini's group has shown that out of thermodynamic equilibrium and in resonance, these can be truly long-range interactions with a 1/r³ scaling. So these have been complemented by experiments in the BSA protein, which is sort of a standard candle for protein chemistry, where these BSA molecules are tagged with a fluorophore Alexa that absorbs in the blue. And when injected with this energy from a blue laser, as well as with a broadband UV LED, we can measure the terahertz vibrations as we go from photons in an optical excitation to a phonon condensate in the protein. So this is the signal that was observed in 2 experiments where the terahertz absorption, you can see this feature at about 0.314 terahertz that shows up in both experimental apparatus only for the solution with marked proteins. And this agrees within 2% with a prediction from Bastrikov in '94 of 0.308 terahertz. So excellent agreement in a classical description. This is not a quantum effect. But what is a quantum feature, if you can see there we go. What is a quantum feature is the enhancement of the absorption peak. And Marlon Scully's group at Texas A&M University published this in 2019, showing that the time scale for the quantum coherence of these collective phonon oscillations and the deepening of that peak goes from quasi-thermal statistics to super Poissonian statistics. And the quantum coherence length for these phonons is about 5 microns for this system. So we wanted to understand how these simulations or how these experiments could occur in this complex system. So we have simulated this with Ali Hassalani at the International Center for Theoretical Physics and Hatere Azizi, our postdoc, to look at optical excitation of these tryptophan chromophores that are in the BSA complex. So this is without the Alexa fluorophore and in equilibrium, but we simulate this huge water box of about 13 nanometers in size. And we wanted to see if we could see sort of allosteric mechanical restructurings as a result of these optical excitations. So we were able to do this using supercomputers at Oak Ridge and Argonne National Labs. So these tryptophan chromophores, we get this from high level density functional theory calculations and beyond. But then looking at those charge distributions, we can then see mechanical restructurings of amino acids that are near the tryptophan, but also far. So this is just showing the change in the free energy landscape of the tryptophan with excitation. And then I can show you these restructurings as maintained in the terahertz spectra of the BSA water environment. And the key point here is that if you look at the tryptophan next to, let's say, its nearest amino acid lysine, you see these features in the difference between the ground and excited state spectra in the few terahertz regime. And then as you go out into the entire protein, determining the cross correlations, you get a factor of 10 increase in the absorption. And then as you go into the ions, you get another factor of 10 increase in that few terahertz regime. And then as you go into the water environment, again, with the larger number of water molecules, you have sort of this nonlinear terahertz cascade into the water environment in the tens of terahertz. So this is a very interesting finding that we need to get more statistics for. But we're looking at applying this as well to non-aromatic fluorescence in amyloid aggregates and glutamine crystals, which have exhibited this type of fluorescence behavior, even in the absence of aromatic chromophores. So I'll skip this for now, but just to highlight that looking at quantum nuclear effects and how they couple to plasmonic excitations is a current very active area. And so looking at, say, these representations of something similar to amyloid fibrils, which are implicated in neurodegenerative disease, we can actually show that these MBD modes actually are influencing these nuclear coordinates and vice versa. And so we ultimately, what we'd like to do is to get to the point where we have forces from these plasmon modes that actually project forward the nuclei. And so we can do that in a sort of consistent way with Hellman-Feynman. But looking at these characteristics of even these small proteins, we see this similar behavior, the similar correlation between the these entropy peaks and the density of states of these modes. So just to conclude, we talked about plasmonic allostery and these quantum fluctuations to be correlated for allosteric synchrony. We looked at superradiant excitons and the native symmetries in these biopolymeric aromatic networks. Out of equilibrium is key in biology, and we can see that out of equilibrium environments generate not only these giant dipoles that exhibit phonon condensates, but the photoexcitation of residues can be communicated to the environment in this terahertz regime. And finally, I just want to thank all the people without whom I couldn't do the work that we do, and particularly the people that I highlighted in this presentation. So thank you very much for your time and for your interest in this exciting area. Thank you.
Thank you, Philip, for this very nice talk. I would have a question.
Sure.
What do you see of some, from your perspective, what sort of experiments are needed in order to test all the hypotheses that you presented to us, all the theory that are so beautiful and that you presented to us?
Sure. So let me go back to this. So for plasmonic allostery, right? So what we're doing actively now is trying to find the coupling between fragments of your complex system, because in biology, in terms of how biologists and biochemists see it, they see it in terms of subsets, subsystems of your entire complex system. So one experiment that we could do is to figure out a way, if we see a signature of entanglement or highly quantum correlations between two fragments in your protein DNA system, we could envision a way of transducing that kind of quantum entanglement to a photonic readout. And there have been some great experiments where we're working on developing with Prem Kumar at Northwestern University, which he showed in 2017 that green fluorescent protein can actually be a sort of DBIO crystal in biology, which produces spontaneous parametric down conversion of a single photon into two entangled photons. Could we possibly use that as a tag to transduce that to a readout? So that's one type of experiment. The experiment with, let's say, ultra-fast UV spectroscopy could be a transient absorption experiment or a fluorescence upconversion experiment in the microtubule. It's been done, Majed Chergui showed in Science in 2013 that you could show this coupling between individual tryptophan chromophores in the myoglobin protein. And so we could envision doing this in the entire microtubule as a signature of this superradiant shift that occurs in the whole system. And then the experiments that are currently being done in Montpellier and in Rome and also we're developing them at Howard, is to look at how this terahertz signal evolves with changes in the protein water environment. So as you photoexcite, are there other ways to excite this collective phonon condensate? It's been observed already in 2018, it was published, but can we excite these residues in other ways, let's say through the ATP transport or electron transport change? And can that be a way to then communicate this information in the aqueous environment? I hope that answered your question.
Yes, thank you very, very much. It was very nice having you. And now it's my pleasure to introduce Judith Klinman from Berkeley, who is going to be presenting, representing Adam, with whom she worked with in the past. And Adam has had a personal problem that he cannot attend, but his slides will also be available when we upload everything to a place. So thank you, Judith. Can you, we can see your screen. I'm not sure if we can hear you.
Me? No?
We can hear you now. We can hear you now.
And you can see the screen.
Yes.
Okay, let's go for it. Good morning or good afternoon, wherever you are. And I'm glad to be able to help out in this way. I am Judith Klinman, and I'm going to be talking about quantum tunneling and new ways to understand enzyme function. This is something we've been interested in really for decades. Let's see where we are now. So the question has, in our minds, is related to biological function. The specifics are enzyme reactions, and this is an area of great interest for fundamental reasons and also because of green chemistry and many applications. Theoreticians have been trying for quite a while to see if they can't design de novo enzymes that will catalyze reactions at very rapid pace. And what I'm showing you here is a plot of how often the approach succeeds versus the rate acceleration. So in 10, you see the consequence of ongoing efforts using computer design to achieve proteins that catalyze reactions about 10 to the 7-fold faster than the comparable reaction in solution. That's one way to approach it. We all know about directed evolution, pioneered by Francis Arnold. And if you take these computer designed proteins and then apply biological evolution, you may get up to a rate acceleration of 10 to the 10-fold. However, actual degree of rate acceleration is seen in red, and we know the biological systems work at rates that are 10 to the 20, actually up to 10 to the 30-fold faster than the comparable reaction in solution. What is missing, and one issue we're extremely interested in, is motions within the protein, how we relate these to catalysis, and how we describe them. So quantum mechanics is a powerful tool, and it has provided us with a new way of looking at biological catalysis. And a good way to look at this to start out is the de Broglie wavelength. We're going to focus on nuclear tunneling eventually, but most biologists have examined electron tunneling, primarily in the literature until recently, last 10 years at least. And the reason that electron is such a wonderful quantum agent is that its wavelength, if we just look at the computed de Broglie wavelength, is in the order of, let's say, 30 angstroms with a particular input of 4 kcals per mole. If we move to the nuclei, proton, deuteron, tritium, carbon 12, you see a corresponding increase in mass, of course, and a corresponding decrease in de Broglie wavelength. But the important issue is the de Broglie wavelength in relation to the reaction that's being examined, and it turns out that in hydrogen transfer reactions, the hydrogen is going to move in the range of 0.5 to 0.7 angstroms. Bingo, we would expect quantum behavior. Now, it's been recognized in the literature for many years that hydrogen can tunnel it, and using carbon-hydrogen activation as model systems where you can stably incorporate isotopes into the hydrogen bond, protium, deuterium, tritium, you can look at the rates, you can look at the rate ratios, how fast is protium versus deuterium, and you can look at the temperature dependence. And it's been known for many years now that if you go to low temperatures near absolute zero where there's no thermal activation, you get, in fact, very large isotope effects, and rates proceed via this quantum phenomena, but without any effective energy of activation. So this observation of similar slopes and virtually no energy of activation is diagnostic of hydrogen tunneling at low temperatures near absolute zero. Now we're interested in biology, which takes place under the warm conditions of life. And can we see anything that is similar? And if so, what are its properties? And on the left, I'm showing you an idealized slope of Arrhenius plots where we're once again comparing rates for protium and deuterium labeled substrates. And what we have, what has emerged over several decades of work now is the appearance of slopes that within experimental error and within the biologically accessible temperature regime, these slopes are the same, i.e. they're independent of isotope. This is contrary to semi-classical considerations, which would look more like the panel on the right. Now, the thing about these data shown in the panel on the left is one, that the energy of activation appears to be identical for protium and deuterium. But the twist is that the slopes are not zero. We're working under biological conditions where there are always some thermal activation processes thermal activation processes dominate biological reactions. The other twist is when we make impairments to our enzyme systems, we see a transition to the panels in the panel in the middle and on the right, where these lines start to converge, approximating what would have been diagnosed as semi-classical behavior. And then when you get even more impairment, the lines start to cross. Okay. Now, theoreticians have for a long time wondered how to explain this. And we've been very fortunate that this enzyme, lipoxygenase, has given us a set of data which have allowed the development of very robust non-adiabatic quantum tunneling theoretical formalisms. And this is an enzyme that works on fatty acids. Fundamental for today, we pull a hydrogen atom off of a carbon in the middle of two double bonds. The proton goes on to the hydroxide of a ferric hydroxide cofactor, and the electron goes on to the iron. We convert hydroxide into water and ferric into ferrous. And this step has been isolated and studied in detail. In the inset, we see the active site. Here's the ferric hydroxide. Here's the bound substrate, long fatty acid. The black is the bond that's moving from substrate to donor to ferric hydroxide acceptor. And we see also that there are fundamental residues that are conserved around the reaction site. There are two hydrophobic residues, Lu546 and Lu754, that encompass the reactive CH bond sitting on either side, and another hydrophobic residue, Ilu553, which is one helix turn away. On the right, you see the results of the study. And what I want to call your attention to is in red, that if we look at the difference in energy of activation between the heavy isotope D and the light isotope H, we see that there's a small difference, not zero, but close to it. And then as we make changes just in the volume of the Ilu553, going from the native Ilu to valine, leucine, alanine, glycine, we're making bigger and bigger holes. We're deleting the volume and making bigger holes. And look what happens. There's the transition in the delta EA, or difference in energy of activation. So the theoretical approximations are as follows. We need multiple dimensions to understand what's going on. And in the left, you see a breakdown into the different coordinates which have to be considered to approximate this behavior. We have an enzyme which in its enzyme substrate complex will exist between two states. We've seen this through a number of approaches. In fact, in SLO, we have an inactive state and we have an active state, and everything will be moving around. There'll be some equilibration. And then we also have some highly activated state where things start to get better aligned and closer together. And that's just represented as A star. Now, at some point where, in fact, you are positioned correctly for tunneling, the overlapping parabola pictorial representation developed for electron transfer kicks in, where what we're doing now is putting in the thermal excitation, which is needed to bring the donor and acceptor modes into degeneracy transiently through this thermal activation. That's called the tunneling ready state. And you can see this is the point at which you get very effective wave function overlap. So the thermal activation comes dominantly from the energy needed to transiently achieve this tunneling ready state. Now, this tunneling ready state is approximately 2.7 angstroms. Of course, van der Waals distances are about 3.2. We'll get back to that in a minute. But the thing about this coordinate, A, B, and C, is that we have separate coordinates for the temperature dependence and the isotope dependence. So according to this representation, the activation is temperature dependent but isotope independent. The wave function overlap itself is isotope dependent, but temperature independent, of course, because it's a quantum movement of the particle. And this actually predicts temperature independent kinetic isotope effects, which is what we see for our native enzymes. However, hydrogen is not electron, and small changes in difference can have a big effect on wave function overlap for the nuclear particle, much more so than for the electron. And that's shown in the gray box, where the tunneling rate takes the static picture. And now we're going to integrate the sampling over many distances. And when you do that, you get, if the protein is already well positioned, the original state, where the lines are parallel, delta EA is about 0. But once we perturb the enzyme and the donor and acceptor move away, which is what we see over and over again, now all of a sudden deuterium starts to encounter more difficulty than protium because of its smaller wavelength in achieving a tunneling ready state. And this leads to a disproportionate increase in the enthalpy of activation for D over H, and there is our delta EA much greater than 0. So the combination of these different coordinates has been extremely successful in allowing us to at least propose a working model for understanding warm conditions, room temperature, biological tunneling of nuclei, not the electron, which was previously well explained. Now, what the tunneling ready state was 2.7, let's say 2.8 angstroms is an outer limit. We did not have an X-ray structure for the complex of SLO, lipoxygenase with its substrate. And we turned to a collaboration with Brian Hoffman, who was instrumental in developing Endor measurements, where we look at electron spin interactions to look at, for example, bound substrates at enzyme active sites. So we are now looking at SLO, where we're using manganese plus 2, as a placeholder for ferric, which is the active form in SLO. And we've labeled the substrate with carbon-13, and we're going to look at nuclear spin on the substrate and electron spin on the cofactor interactions. The next slide summarizes quite a bit of work over many years, and these are the distances that we see between the reactive carbon of the labeled linolenic acid at C11, and that's the donor of the hydrogen, and the oxygen is the acceptor. This distance is 3.1 angstroms in one of 2 configurations that the Endor studies detect. The second shows a longer distance of 3.8 angstroms, and we conclude that A is the reactive ground state, van der Waals distance conformer, that will lead through a variety of different conformational sampling states and the environmental reorganization in the Marcus equation to lead to the required 2.7 to 2.8 angstroms. I just want to show you a new result. It just came out in biochemistry, where we actually took the same series I referred to earlier, the 553 Aiello, and generated the mutants seen above here. And we then looked at 2 parameters. One, we did the Endor experiments to ask what fraction of the enzyme can get into this active A conformation as a function of mutation. And when we did that and compared it to our previously determined increases in delta EA, bingo, a straight line. And the relationship between delta EA and the equilibrium donor-acceptor distance before this donor-acceptor distance sampling coordinate is shown on the right, it's close to the 2.8 in the wild type, not much distance sampling needed. It gets further and further out as we make the mutant, and concomitantly, we have to incorporate the donor-acceptor distance sampling. This kind of result is really powerful confirmation of the tunneling event itself and the theories that have been developed to explain it. So let's move on to the relationship of the protein itself to what we're seeing. These are really exciting days. And one question is, okay, if we're at van der Waals, how do we get to 2.7 Angstrom for the tunneling event to occur? This is a temperature-dependent process. What's the enthalpic barrier? And our model, which I will tell you a little bit about today, is that it's the protein scaffold that is co-evolving with active site chemistry. Now, with Adam Offenbacher, who was a postdoc in my lab, and before moving into his independent position, we began to explore physical methods for looking at the transmission of thermal energy in proteins. And of course, there are all kinds of ways you can think of. One very powerful technique in principle is room temperature X-ray crystallography. But the only method that turned out to be profitable for lipoxygenase and now many other enzymes is to do a technique called hydrogen deuterium exchange linked to mass spectrometry. Essentially, we take a protein, it's breathing all the time, local unfolding. We put it in D2O, we let it go for a little bit of time, we quench at low temperature and pH. We break the protein into many peptides of average length, let's say 12 amino acids, and then we look at the time dependence of deuterium incorporation. It's how long we let the enzyme sit in D2O. And we, importantly, which is quite different from what other people have done, we do this as a function of temperature as well. The data we collect are under a condition called EX2. We're looking at the opening and closing of the protein followed by a slower exchange of the peptide bond protein by deuterium. And what we're looking at then, primarily when we change temperature, it's a combination of both the K open over K close and K intrinsic. But importantly, what we're going to do is mutate the enzyme, as I showed you earlier, and redo these experiments with a mutant, and we assume this doesn't change. And what we're able to do is pick up the temperature mutational dependence of this big K open, or the ratio of the opening and closing of the protein, through this local and transient unfolding process. Now, just cut to the chase because time is short. This is the entire structure of lipoxygenase. The iron is shown by the orange ball. The residues that are highlighted are going to be shown as space filling. And here I'm just pointing out I553, which is the series we've been looking at in this talk today. And we picked out of all the peptides that we analyzed, which gave us close to 100% coverage of the protein, only 2 of them showed a temperature dependence that was further sensitive to mutation. And here we are looking at, once again, the wild type, our 553G, which I've already talked about in the context of kinetics, and another mutation, which is L546, which we converted to an alanine, which is one of the 2 residues that sits on either side of the reacting CH bond. And what we found was this incredible correlation, which is the energy of activation for this local opening and closing just in this region of 2 peptides that go from region 284 to 334 in the amino acid sequence show the same trend as the energy of activation that we saw previously for catalysis. The magnitude of the energy of activation is different. We're looking at different processes, but the trend is there. So where is this happening? This is showing you the solvent protein interface going into the active site with these 2 peptides. So where is this thermal excitation happening that we're picking up by HDX? And here it is. This is a spatial network that shows us going from the protein solvent interface into the active site. And it is comprised of all hydrophobic residues except for a single hydrogen bond, and contains the 2 hydrophobic side chains I just showed you, 546, 553. The model then is solvent comes through its kinetic energy, interacts with the bound biological water, and leads to thermal excitation through transfer of energy from the bulk water. Now, the HDX experiments I've just described are good for, they're not just good, we think they're excellent for spatial resolution of what we call thermal conduits. But what we wanted to do was move into real-time measurements. And this is a picture from a paper published by Bakshi in India quite a few years ago, where once again we talk about the protein with the bound water, some free water in a region called biological water, and the bulk water. And Bakshi was looking at the rate of exchange of structure and energy between these different classes of water, and described them in terms of either picosecond or for the bulk water, or nanosecond for the biological water. Well, we decided to once again go back to SLO, our model system, and try to get direct evidence for communication between the protein surface, which will comprise a protein-water interface, using a fluorescent probe called BEDAN, which we attach covalently in two places in lipoxygenase, either on this loop, which is on the tip of the thermal conduit we identified by HDX, that's 3Q322C BEDAN. And we also put the chromophore onto a control region called 596, which is far away, and through the HDX experiments indicated no dependence of its thermal activation or mutation. Now, these are nanosecond Stokes shifts, and below you see a summary of the energy of activation for each of these processes, where we look either at the environmental, it's a solvent environmental reorganization around the excited state chromophore, where the BEDAN is on the thermal network, that's in red, or in the remote control position. And we can see very nice agreement between the energies of activation for the Stokes shifts when we put the BEDAN in the thermal conduit position, but no sensitivity in terms of temperature dependence in the control position. So we now think in real time, we're actually measuring thermal transfer. It's happening, of course, on the surface where the chromophore is, and involves reorganization of solvent around the surface. And we find that it's quite positional specific. But the shocking thing in a way is that the thermal activations are the same. Maybe it's not shocking if we had thought more carefully in advance, but we continue to be surprised by the data that emerge from these studies. So essentially what we've seen is a series of studies, and what I just talked to you about today are the anisotropic protein quake that we're seeing at the BEDAN in SLO, which is occurring on a nanosecond time scale, and which has the same enthalpy of activation as CAT, which is happening on a millisecond time scale. We also have a study I don't have time to talk about on a thermophilic dehydrogenase, where we're transferring a hydride ion, where we see a quake in a way, but it's an internal quake. We look at fret between an internal tryptophan and the nicotinamide ring of the bound cofactor, and we see a microsecond transfer of energy into the protein, which allows the fret to occur. And this also occurs with the same enthalpy of activation as what we've seen for catalysis. So how do we explain conserved activation energies with all these rates, which are all over the place in terms of time? And that's where this rate constant that I've summarized for you is fundamental. And it's basically that any observed process that we are looking at has the intrinsic process with an intrinsic enthalpy of activation, and that's multiplied by an F conformation in a protein, where this is the probability that that collision of surface water, let's say, with the protein will lead to a productive phenomenon. So in catalysis, it's very hard to get everything organized, and F conformation is quite small. We've already seen that through theoretical analyses of tunneling reactions in proteins. A T-jump, where we have to organize the donor and acceptor, may also have a lot of requirement to get the geometry just right, and F conformation is still small, although larger than catalysis. We go out to the solvent now, and the need of reorganization, the fraction of time we get the right configuration for the Stokes shift to kick in, is more probable, so the reaction is faster. And we've already, in the literature, seen examples where the actual heat transfer itself through the protein can occur from femtosecond to picosecond. These are direct measurements where you photoexcite a bound chromophore and look at the heat flowing out into the solvent. So we have this very rapid heat transfer with similar enthalpies of activation for related processes, but different probabilities that they will occur, and hence a different range of rate constants. So we think that we are moving to a picture that allows us now to move forward, and that has to do with the idea of protein anisotropic evolution, which links the protein solvent surface to the active site and provides for heat transfer, and does it in a way now where we need direct measurements in these networks to see the physical processes that describe the networks themselves. So I just want to finish up with acknowledging the people who, many people have been working on SLO and other systems, and just to highlight the key players over many years for SLO, Mike Knapp, who developed was the first one to develop this non-adiabatic theory, Adam, who did the HDX experiments and the beginning of the Stokes shift experiments, Paulo Zaragoza, who has done the recent Stokes shifts and their temperature dependence and mutation dependence, which I didn't get a chance to talk to today. These data are in process. And then I want to acknowledge all of our collaborators, in particular theoretician Sharon Hamish-Schiffer, who has taken our initial theory, developed it considerably further, and Brian Hoffman, who was irreplaceable in the application of Endor to describe the ground state structures in SLO. And thank you very much. I'm glad to be able to be present, even though I wasn't originally planned to be at this meeting. Okay.
Thank you, Judith. There is a question from the audience.
Yeah.
I am not sure to which slides it refers to. It says, what is the enzymatic rate acceleration time scale here?
The enzymatic rate, so the enzymatic rate is millisecond. So in general, all enzymes converge on a single rate constant, pretty much. I mean, obviously they have orders of magnitude differences. Some are much work per minute, some per second, but millisecond is the time regime where biology functions in this way. And so we're looking at, in relation to the type of physical measurements I described, much slower processes than occur with the sort of thing we see through Fred or Stokes shift. I don't know if that answers the question. Yes?
Another question is from the same person. We
Okay.
know that the tunneling rate is inversely proportional to mass. So how could we look at proton versus electron tunneling rate?
Okay, so I think when we get to tunneling, unless you've thought about it deeply, and it's a very deep subject, and Marcus theory really has taught us that, the process of tunneling itself is believed to be instantaneous. So if you talk about either electron or proton tunneling, it's the environment that controls the tunneling rate. So if in fact we have an environment that facilitates the environmental reorganization, and in the case of protium, the donor acceptor distance sampling, the rates can be as we measure them in real time. So for example, in the SLO reaction, where the hydrogen is moving in a rate determining step completely via tunneling, the rate constant is 250 per second for the wild type, and then it goes down as we make mutants. So in the case of electron transfer, of course, unless you're looking directly at electron transfer, it's hard to say. In many ways, SLO is another good example because it's been treated as a proton-coupled electron transfer. So if we think of both the proton and the electron tunneling, once again, these combined processes are happening on the time scale of a millisecond, not instantaneous. And that millisecond delay or slowing is due to the fact that we have to get this thermal transfer into the protein. And what I'm talking about today is a way of starting to understand how this thermal transfer takes place.
And Judith, now I have a couple of questions of my own.
Okay.
My question is, have enzyme people like yourself started trying to see whether and how enzymatic activity might depend on other spin phenomena like the existence of radical pairs or this chiral-induced spin selectivity and so forth.
What was the last thing you said? I couldn't hear you.
Chiral-induced spin selectivity.
Oh, yeah. Well, certainly the radical pair issue is a very interesting one in our view, because in fact, I mean, basically what we're getting is long-lived coherence. And so the question is how do we think about coherence in the kinds of enzymatic reactions that I've been referring to? And to us, the coherence is this transient energetic degeneracy, which allows the hydrogen to move back and forth as a wave until you break the degeneracy. And so we don't know the lifetime of those degeneracies. We know the tunneling is essentially instantaneous once it's achieved. What we think is that these transit degeneracies have very short lifetimes, but we have repeated thermal activation, and that's how you get the reaction to proceed. So the question then is, how do we think about this going forward in terms of overlapping disciplines? We've been thinking a lot about the presence of these thermal networks as a means to generate this transient degeneracy. But of course, the degeneracy is short-lived. So I don't know how we could sustain the degeneracy via these thermal networks, because in the case of the electron pair, once the unpaired spins go into a paired spin, you'd need subsequent activation to get them back into the unpaired configuration. So maybe there is a dynamic effect going on, such as we see with hydrogen, where we get into degeneracy and it breaks, and then we get into degeneracy and it breaks. But I think if these are thermally activated processes, these thermal networks may be a really useful thing to pursue going forward in terms of trying to generate coherence and control coherence.
Thank you, Judith. More questions. We have 3 minutes. If someone wants to quickly unmute themselves to ask questions directly to Judith. Well, Judith, thank you very much for joining us today. And I really hope that we can continue the conversation.
Okay, great. Enjoy the rest of the symposium. Thank you
very much. Thorsten, are you ready to?
I am ready, yes. So let me share the screen. Okay. Can you see my screen?
Yes.
Yes, okay. All right, so thanks for the invitation. And what I want to do is, since the early days of magnetic sensing and quantum biology, in particular, magnetic sensing has always been part of the discussion because of this idea that there could be a spin coherent mechanism involved. However, that question of how birds and other animals sense the magnetic field is rather old. We know it since about 50 years ago, where the pioneering experiments of the Wilschkos showed that if you put a migratory bird in a cage, it will hop north. And if you then change the magnetic field, then you can change the direction in which it's hopping. It's been 20 years ago that I suggested a particular molecule, cryptochrome, as a potential magnetoreceptor candidate. And we still don't know whether that is or is not the right molecule. And so I thought it may be worthwhile every 20 years or so to step back and ask, are we asking the right questions? And one of the questions that remarkably has not been asked is, what is the threshold for magnetic sensing in biological systems? I would argue that that's an important question, because when we look at the threshold, often we realize what are the problems, the challenges that nature needs to overcome. And if we figure out how nature overcomes these problems, we may be led to the receptors. I'll give you some ideas, some examples. If a bacterium wants to detect a chemical gradient, a neutron gradient, it has a problem. Again, if you look at the physics side of things, the number of nutrients on one end of the bacterium and on the other end is essentially the same. The gradient is too shallow to have a big difference there, or actually different larger than one. So even if you have super sensitive sensors and receptors, it wouldn't help you. What you need is you need to have an integrator, and you need to actually make this bacterium swim long enough in one direction to allow it to integrate. And that's exactly what it does. You get a chemical integrator. So here, what nature did to make this work has nothing to do with the receptor signal to noise. It has everything to do with the feedback mechanism behind. A contrary example is vision. Our visual receptors in the eye can detect a low number of photons, that's the threshold, perhaps even a single photon. And the molecule is known, it's retinal, it's a carotenoid that changes shape upon light absorption. Now, in order for this to work, what you need to make happen is that you essentially have no dark noise, that this molecule does not spontaneously ever change shape. And that is achieved in nature. It found a molecular motif where this molecule, this retinal, is essentially embedded in kind of a cast, a salt bridge that it needs to break. And that has virtually no dark isomerization noise that happens once in 3,000 years. And that is the key to make this work. And I don't think we would understand why this particular molecule is chosen if we didn't ask the question of what the visual sensitivity threshold is. You can, if you have like full light conditions, you can have many other things in photosynthesis that absorb light, but all visual receptors in every organism, from the fly to humans, use this one molecule. It's exactly one solution that nature found. And I think that's typical. The more difficult the problem is, the less likely it is that there are hundreds of different solutions to it. So what about magnetic sensing? What can we say about the threshold? Now, as I said, this has not been systematically studied, but we can glean some estimates from 3 sets of experiments. First of all, often people say that in the magnetic compass, we're detecting a 50 microtesla field. That's, however, not really what happens. It's not the magnetic field is off in one situation, then 50 microtesla on in the next situation. What you have is you have a background or the operating point of 50 microtesla, and that operating point can actually be lowered, has been shown to up to about 10 microtesla. But then what the signal actually is a small change around this operating point by turning the head by a few degrees. And if you just think about the sines and cosines and the components and how much they would change by such a few degree turn, you're getting on something of the order of 100 nanotesla or less. That is the actual signal, that's the actual change that you get. There are experiments that actually try to estimate an angle. And so what you do is you take an animal that is in a pond or in some environment where it likes to be, and then you move it by a certain distance, and it homes back to that pond or that location. So this is a study with spiny lobsters here in Florida. And so you move them a few hundred kilometers each way, and then they will respond by swimming back to their original site. And you can do this homing behavior in a lab virtually by instead moving the lobster, actually taking the lobster and then giving it different magnetic fields that it would encounter at these different points. And this has been done fairly systematically by the Lohmann group in North Carolina over 20 years. And they showed that you can change the intensity and you get this response. You can change the angle of the magnetic field with the horizon, the inclination angle, and you get this response. And again, if you estimate or measure the intensity changes, we're talking about on the order of 100 nanotesla here and changes of a few degrees that have been measured. There are a few studies with newts that suggest that this homing behavior could occur over distances of even just a few kilometers as opposed to hundreds of kilometers. And on that scale, we're now talking about angle changes of less than 1 degree or intensity changes of 10 nanotesla. So we again have this 10 to 100 nanotesla range. Now, about 15 years ago, I started a set of experiments where we took the static field and did a normal magnetic orientation experiments and added a very weak oscillating magnetic field. And the motivation was to see, can we actually induce spin flips? Is there a mechanism that is based on this? But nevertheless, regardless of the motivation and the interpretation of these experiments, they just yielded data. And one of them that it shows is that at a particular frequency, which is the free electron number frequency, so just G mu Bohr times the intensity of the static field, we can go down to somewhere around 10 nanotesla and still disrupt the compass of birds. So at 5 nanotesla, we get normal orientation. At 15 nanotesla, these birds are disoriented. The nice thing about these oscillating fields is that in many orientation experiments, you don't want to mess with the static field so much. So this gives us another knob to turn to look at what the threshold of sensitivity might be. Now, when this experiment came out, people did not want to believe it, that this can happen. There have been a few follow-up studies, but then there was one study that couldn't reproduce it. And so for a long time, there was a lot of discussion about it. But since then, there are two other groups, and now 3 groups in total, that have independently reproduced this result that on the order of 10 nanotesla effects can disrupt orientation. And in fact, one group showed in their birds the threshold was at 2.4 nanotesla. That's where they saw orientation, and at 0 knots, so somewhere below 2.4 nanotesla. So the threshold here for radio frequency effect seems to be on the order of 1 to 10 nanotesla. Now, this is, of course, a disruptive effect. So how do we know that magnetic sensing has anything to do with this, that these effects actually affect the magnetic compass? But let's think, that's an objection, but let's think through the logic of that objection. The fact is that birds have some molecular system that's sensitive to 1 to 10 nanotesla radio frequency fields. So now we have 2 options. Either this super sensitive system is not used for magnetic sensing, but just sits there for something else and just happens to disrupt magnetic orientation when it is activated by magnetic fields. The other option is that these receptors, these supersensitive receptors, are actually the molecules that are involved in magnetic sensing and orientation. There are quite a few people that favor option 1. I personally think that option 2 makes more sense. And we've done a number of control experiments that actually support this. So one of the things you can do is you can change the angle of the radio frequency field with the static field, like you would do in magnetic resonance. And we know that if the oscillating field is parallel to the static field, you shouldn't get an effect. And indeed, even if that field is present, it does no longer have any effect on the bird. So we already know that whatever this does, it in some sense is linked both to the direction of the static field as well as the effect of the RF field. We also see that orientation is not difficult, it's not easy to make happen, so it can be disrupted easily. But we can show that there are other responses that are not affected by these RF fields, other orientation responses, again strengthening the case that this is specific to magnetic orientation. And finally, we know that this is a resonance at the Lambda frequency, so we can change the static field intensity. And in this case, we do no longer, so before at 1.3 megahertz, we had this strong effect. And if we change the static intensity, we don't see that effect anymore at 1.3 megahertz. Again, arguing against some non-specific effect where we make the birds nervous. However, what we really need to do to settle this debate is we would need to have an experiment where we take away the ability of a bird to detect the static field for orientation and make it orient to a 10 nanotesla RF field. If we can do that, then we can show conclusively that this is really involved in magnetic sensing and in magnetic orientation. We have some idea how to do this, so stay tuned. We'll try these experiments hopefully this summer. Now, the summary of this brief estimate is that we have a threshold of magnetic sensing on the order of 10 to 100 nanotesla, which leads me to this question of how can animals detect 10 nanotesla magnetic fields that are used as the title of this talk. And I think this is the question we actually need to address and successfully answer if we're serious about finding the magnetoreceptor. The problem is that no mechanism, regardless of whether you believe in magnetite or the radical pair mechanism or induction, can explain this in any satisfactory way. So that's maybe why we shied away from answering this question. It's kind of like it's been difficult enough to detect, to understand how to get 50 microtesla magnetic field effects in biological systems. So, you know, 3 orders of magnitude below this, that's hopeless. But that's a little bit like the anecdote of the drunken man who looks for the dollar under the light because it's only where he can search, as opposed to going, you know, where he lost it, somewhere in the darkness. So I think we should maybe not be afraid of this darkness and go in there. What needs to happen in order to explain this? Well, let's start out, you know, the radical pair mechanism has already been introduced by Martin Plenio. So we have an electron transfer, then we have 2 different nuclear spin environments that set up a stage for then a different effect, differential effect of a static field on this. And so Peter Hoare has done this nice demonstration that even without an external field, you would get these fast oscillations. And then with an external field, you now have a beating of this oscillation that's overlaid, and that depends on both the intensity and direction. And then you can just integrate over this to get the yield, and that's the basis of the compass idea, which was originally suggested by Klaus Schulten. But if we're thinking about the thresholds of sensitivity, Klaus Schulten's original idea is actually a mechanism for a 1 millitesla magnetic field, because he was thinking of shifting away through the magnetic field the triplet states from the singlet state, and therefore getting it out of range where they can mix, and the mixing is done by the hyperfine interactions. So essentially these effects should only occur, the magnetic effects, once you have magnetic fields that are stronger than the hyperfine couplings, and that's typically on the order of millitesla. Peter Hoare showed us, and Chris Timmel, about 20 years ago, that if there's a symmetry where you now break essentially degenerate eigenlevels, you can have a low field effect, and there's similarities in solid state effects of this nature, where essentially you open new channels of singlet triplet transitions through this symmetry breaking, and that allows you now to actually explain Earth strength magnetic field effects. So in both cases, before these ideas were suggested, people did not want to believe that this can happen. In fact, in Klaus Schulten's idea, if you just ask what is KT, the interaction energy of magnetic fields on the electron spins compared to KT, we're 4 orders of magnitudes below KT. So this should completely be buried in noise. But since this is a non-equilibrium effect, you can work around it. That's the workaround that Klaus showed. And then there's a workaround, at least for, you know, getting down to Earth strength magnetic fields. We have no idea what the workaround is for 100 nanotesla field. One easy thing would be if the spin correlation time would be extremely long on millisecond scales, then this mechanism could work. But that's very unrealistic, and it just, you know, it's unclear how this can be done. So a couple of questions that people are asking is, or we've begun to ask, and maybe are the right ones to ask, is can there be a way that we don't need this long spin correlation time? Can we actually get a quantum sensor to work faster? And then other questions are, can the symmetries be maintained that are necessary? They play often a big role. And what's the role of noise? How much time do I have, Clarice? Okay, I'm not sure. So let me just, if you can tell me how much time I have left.
Sorry, about 20 minutes, about 20 minutes.
I also have plenty of time. Okay, so then I'll go a little slower. So one of the things that hasn't been really studied is the question of, you know, how fast is this quantum sensing? So we can do these spin dynamic simulations of concrete molecules where we very accurately model what is the angular sensitivity, let's say, for a compass. And then in these simulations, what we can do is we can just vary these spin correlation times in them. And what we can see is that for different molecules, there is a very different time dependence on these spin correlation times, meaning that if I reduce the spin correlation time in one case, I almost immediately lose this directional sensitivity, whereas in the other case, I'm much more robust against such changes. And so maybe by understanding how you can make this happen, we can understand how to build a quantum sensor that can operate even if the spin correlation time isn't terribly long. Now, when we ask things like what is the directional sensitivity, even after 20 years of research, there's still surprises. So for the longest time, we thought that the typical optimal singlet yield, or the angular dependence, is the sine 2 theta type pattern. But then Peter Hoare, about 3-4 years ago, showed that you can actually have needles where you have an extremely sharp angular sensitivity, but that requires, again, very long spin correlation times, upwards of hundreds of microseconds. So one of the things we understand where these things are coming from, we can look at the energy eigenstructures and the avoided crossings, and Jeff Canfield a long time ago had actually studied these things. And we actually saw them in our original 2000 paper, but they were so small that we didn't do much with them because we didn't assume that we could have spin correlation times more than a microsecond. Now, one of the things that we asked is, what happens if we now introduce a little bit of noise? And in this case, we're allowing the hyperfine parameters to change a little bit from one copy to another and have some variation. And our initial expectation was that if you introduce this amount of noise, it should possibly wipe out this needle. But what we found to our surprise is that the opposite happened. So this blue line here is the needle for as in the Peter Hoare paper, and it's now over here. And now we're adding, essentially what we're taking are the hyperfine parameters, and we're just allowing them to vary within 1% up to 10% around the values that they have. And you see, as you do this, this needle gets much, much deeper. And another way to plot this is if we look at the sensitivity, some measure of the optimality, you can actually see the needle here gets deeper, but it also gets broader. So in some sense, it's not as good for directional sensitivity. So if we quantify this, what you can see is that with this additional noise, you can now actually have a mechanism where originally you needed to have, so this was 100 microseconds spin correlation time, but now you can get the same sensitivity for 1 microsecond. So again, there is a way, even in this classical mechanism of the radical pair, to maybe push things around and work around this constraint. There is another very new idea by Daniel Kapnick, where he essentially says, if we have not a donor-acceptor system, but in addition a scavenger, we could get much quicker magnetic sensing. And in a way, what this is in the Zeno effect, essentially you restart a quantum measurement over and over and over, and the scavenger plays that role to make that happen. And by this restarting over and over, you can actually then get bigger sensitivity even for short spin correlation times. So this is a promising idea. Again, this is all at a very theoretical stage, but it may be that that's exactly the type of problem that we need to address and answer in order to understand how can we get a 10 nanotesla magnetic sensor. Now, on the molecular side, I mentioned that we suggested cryptochrome a long time ago, and the state of the field currently, after 20 years, is that we know cryptochromes are involved. So if you knock out cryptochromes, the magnetic responses go away, and that has been done in a number of systems. However, that's of course not enough. And what we cannot distinguish are essentially 3 scenarios where either the cryptochrome is the magnetoreceptor or it sets the stage for being a receptor. So it could be that indeed the flavin tryptophan radical pair that was originally proposed is involved, but it could also be that activation of light of cryptochrome that leads to a conformational change will then allow molecules to bind that form, essentially cryptochrome forms a scaffold for the molecules to bind that form the radical pair reaction. So in all of these 2 cases, the magnetoreceptor would be directly involved, with cryptochrome would be directly involved, but it could also be that there's a magnetoreceptor and then cryptochrome is a necessary downstream element from it. And so if you knock it out, you wouldn't see the magnetic response anymore. At the moment, we cannot distinguish between these scenarios, and we only know it's involved in some form. What has been very new is through my collaborator Margaret Ahmad in Paris, and following a Chinese group who did this first, is that we can measure magnetic field effects essentially at the point of cryptochrome protein. The first thing that happens, I mentioned, when light is absorbed, is that there's a conformational change in cryptochrome, and it exposes the C-terminus, and then there are autophosphorylation sites, so there's autophosphorylation happening, and so there's a weight change of the protein, and you can just monitor that through Western blot, and then you see magnetic field effects on this phosphorylation of about 20% change on phosphorylation. So that's a measurement you can do essentially ex vivo. If you do purified protein, you don't see a magnetic field effect. So clearly there's something else needed, partners. But if you just do a protein extract, you see these magnetic field effects. And at this point, the fractionation and other molecular biology hasn't been done to find out what these other partners are, but there's a path towards finding them. The other big thing that happened is that we finally have a somewhat robust genetic system. And intriguingly, it has to do with the clock of fruit flies. So to build a clock, what you have is a transcriptional repressor. So you have a molecule that when it gets expressed, it then represses its own production, and that's, you get an oscillator. And cryptochrome is that molecule is period or pair, and cryptochrome is necessary to bind to pair so that it can enter the nucleus and actually do this repression. And cryptochrome gets destroyed by light, but if you have just a little bit of light so that you don't completely destroy it and thereby short circuit the feedback loop, what you do is you lengthen the period of the clock, and then with magnetic fields you slightly shorten that period compared to this, and it can all be explained in this biochemical way. It makes sense. It has been reproduced independently by two groups, in some sense three if you think cockroaches are similar enough to fruit flies. So that's nice, and it allows us now to study things. And one of the things we've done is to begin seeing whether we actually see these radio frequency effects on these molecular or genetic systems. And we started with fairly large fields to just make sure whether there's an effect at all. We see effects of 2 microtesla RF fields that are off the Larmor frequency in the cryptochrome phosphorylation assay. And in fact, they're very similar to what happens when you just shield the static field. So again, it's similar to what happens in the birds where there's a disruptive effect. And then we saw a 400 nanotesla RF effect. Again, we used a broadband field here on cockroaches. And of course, the program forward is now to see how low can we do. And if we see effects of less than 100 nanotesla at the Larmor frequency in any of these systems, that will challenge the current idea of the radical pair mechanism. There is some other trick, some other thing we need to figure out how it's working. And it may be as simple as that Daniel Kapnick's idea is right, and by identifying the molecular players, we will identify what that scavenger molecule is, and then we can go forward. But ultimately, with this, we have a possibility, both through genetics and molecular biology, to identify the molecular system, the identity of the molecules that are necessary in addition to cryptochrome in order to make magnetic sensing happening. And it's also clear, since you can do this now in a test tube, that cryptochrome alone plus these other players can be enough to build a magnetic receptor, which is a powerful tool to detect what is happening. And what we ultimately need to be able to do is, I think, so this is the program, if we can show that birds indeed detect 10 nanotesla RF fields and they can orient towards them, if we see 10 or 10 to 100 nanotesla RF effects on cryptochrome plus whatever is in there and identify it in a molecular or genetic system, we will then be finally in a position to model realistically with a full-blown spin dynamic simulation on a concrete molecular system what are the magnetic field effects, and answer the question of how does nature do it? How can it happen that you can have 100 nanotesla or 10 nanotesla effect on such a system? And I think that's the big challenge, because at the moment we have no idea how to make this happen. If we can show this, I think then we will have a very clear path forward to experiments to prove this. But at the moment, all we have are very hand-waving, models that are not realistic at all. And I think that's a big challenge. And maybe it is understandable that we can't have a full understanding of this mechanism, because we don't know what the molecules are. We maybe, we're not even sure what the players are in the system. So a lot of modeling may be a bit premature. So that is the path that I think, for the first time in 20 years that I'm in the field, I think if these two experiments actually come out the way that we hope they come out, we would have a fairly clear path towards identifying the magnetoreceptor. Now, obviously, I'm assuming here that whatever is going on in birds is the same as what we are happening here in the molecular system. But going back to what I mentioned with vision, it is remarkably difficult to think about how you can build a 10 nanotesla magnetic sensor in a biological system. I doubt that there'll be hundreds of different molecules that can achieve this. So if we find one, I think it's a very good bet that this will be the one molecule that nature can pick for this. I hope that this presentation will invite others to think about this problem and to take it seriously, how we can get such weak magnetic fields, as opposed to keep modeling how we can get 50 microtesla magnetic field effects on a biological system. So thank you.
Thank you, Thorsten, for this very nice presentation, which, as you know, is super relevant to what we're doing here at UCLA. So I'll start asking questions because I have a couple here, and I know that Philip also will directly ask you a couple of questions. So thank you for mentioning that magnetic field effects might extend beyond navigation, organismal navigation, right? That it can play a role, say, in clock regulation and metabolic regulation. I would like to ask you to speculate a little bit. I know that cryptochrome is particularly sensitive to tiny magnetic fields, right? But could you speculate about magnetic field effects in other proteins that might have the same ingredients as cryptochrome for magnetic field effects? And could you please also speculate on whether light is necessary for magnetic field effects in general in biological systems? Can magnetic field sensing in biological systems be much more widespread than thought?
So going to cryptochrome, one of the things that, the reason we suggested cryptochromes is partly because there were, was a radical pair in them. But the other one that's perhaps more important and has been overlooked, and I need to get the message across better, is that cryptochrome is a biological receptor. It is cycling between an on and off state. And the magnetic field allows you to now change the transition from one state to the other slightly. But that if you have such a cyclic system, what you do is with this change, it gets essentially written away into how much on and off state you have. If you have 1%, faster transition one way or the other, you change that dynamic equilibrium. Very few other molecules that have been discussed actually fulfill that criterion. So it's, if you have something that just switches on and stays on, it doesn't matter that you have some magnetic field effect on the forward reaction. It's not a good receptor. So there's a number of things that are usually not considered because it's so much dominated by spin chemistry, where you're looking at just one reaction, but you need to place this reaction in a meaningful biological context for reception. That's what cryptochrome does. Many other reactions, you would have to now find this, what is essentially the on and off state of your system that does signaling or something like that. And I think that's the main challenge. If I just look at the spin chemistry of cryptochrome as opposed to other molecules, the only thing that's a little bit special about cryptochromes is the spin correlation time seems to be a bit longer. but the actual magnetic field effects are not dramatically better than, you know, other molecules you could look at. It's just you know, the kinetics and it being a receptor, I think, that makes it different. And that's one of the things that has bothered me, because if I see a magnetic receptor, I would like to see a molecule that's in some sense really optimal for detecting magnetic fields. And cryptochrome doesn't really fit the bill, at least not the forms we've looked at so far. Maybe we haven't looked at the exact right species and so on. And so again, cryptochrome could not explain how you get 10 nanotesla effects, at least not in the description we have so far. The second question was about light. And again, that is historical, I think, because it's so much dominated by spin chemistry, where we shine in light to get your electron transfer. You could induce the electron transfer metabolically, and I don't think there's anything fundamentally impossible for this to work. Again, you would have to think about the biological receptor and signaling context. Where does this occur, and so on. And that's, I think, the part that, you know, for me is the make or break very much on this. And that's why I also presented in the beginning the chemotaxis, because for me, it's not even clear whether the receptor itself needs to be special for magnetic orientation, or maybe there's a feedback loop or something behind in the, you know, the biochemical, you know, or neuronal, you know, calculations that are being done. And maybe that's the critical part. Maybe birds can only orient if they fly for long enough in one direction or something and integrate a signal. No, we don't know that, but it's there's a possibility that you could build a sensor that way. And again, it's that context that's been overlooked in my mind, and only now is begun to get looked at.
Thank you, Thorsten. Philip, do you want to?
Sure. Can you hear me, Thorsten?
Yes, I can.
We may have talked about this question, but I thought I'd ask it directly and get your thoughts on this. Do you have suggestions for how we could engineer biosystems to transduce electronic coherences to these longer timescale nuclear spin coherences, where we could then, you know, be measuring kinetic isotope effect and some of the techniques that Judith uses in her lab to observe these longer timescale behaviors, but then to use those electronic coherences? Is there something that you've thought about for suggestion?
So I haven't thought about this question, and it's a meaningful question to ask. It's a good one, but I don't have a bright idea, you know, off the top of my head of you know, what we could do, and maybe it's not so wise for me to just speculate randomly now.
I think Judith may have an answer.
No, I don't have an answer, but I got
Okay.
lots of questions. I'm sorry, I missed the meeting in December also, because there's so much going on here. But Thorsten, you said something really interesting about just small changes in the relative lifetimes of the triplet and singlet states might be enough, because the amount of the small nanotesla changes that can affect the bird behavior or whatever you're looking at. So I'm wondering, I don't know the physical chemistry well enough, but in terms of the environmental interaction between triplet and singlet states in a protein, I'm wondering if you couldn't have, through protein-protein interactions, perhaps downstream through the cryptochrome, I don't know the specific interactions, a local change in the electrostatic environment. So it's transduced through the protein-protein interaction, which can go through over long distances and then change the local environment. And you may just have to tweak it a little bit to move the equilibrium one way or the other. So, and I was really struck by your point about how little change there has to be in order for these effects to be read out. And so that was just a wild off the top of my head thought. And then the other has to do with the relationship between what you call the small changes that are deduced and the coherence lifetime. I'm having a hard time connecting those two ideas. Okay,
yeah, so let me talk about this. So the basic idea of what you're doing, it goes back to, you know, maybe I'll pull up this picture here. You see that going from singlet to triplet, you essentially get an oscillation. And so what the reason you get this oscillation is that if you have an electron spin in a magnetic field, it will precess around the axis of that magnetic field. And that precession time is set by the magnetic field strength. And so the internal magnetic field, this is essentially what you see are these precessions, like each step, each period, you've gone through one precession. And so that sets a time frame. And so if you lose the spin coherence before you go through one of these oscillations, I mean, it's hard to see how you can actually build a sensor out of that detects anything. So that's where, you know, of course, now the time scale that's set by the 50 microtesla field, that is even slower. It's this beating on this here. And so normally there's just a very simple, you know, it's essentially the precession time in a given magnetic field. That should be the minimal spin correlation time you need to have some effects. That at least is sort of the rough estimate. And what I'm saying is maybe that wouldn't work. If you assume that, you cannot explain how these 10 nanotesla effects come about. And so maybe we need to find ways to work around that this is what needs to happen. And some ways you can think about this is it goes back to that symmetry. If you think about a situation where you have some very strong interactions, but these interactions would be the same on both molecules. It doesn't matter that they're there as long as if you maintain some symmetry that gets broken only by the magnetic field. And so you could imagine some scenarios where you set up in a complex system some kind of symmetry that's maintained even though the spin coherence of all this other stuff gets destroyed. But that one is essentially very similar to the decoherence-free subspace ideas that are discussed. But the question is, how would you concretely realize this in a molecular system? What would that be? And that might lead us to you know, some discussion of this. And I'm also wanting to challenge a little bit just this idea that you must have that spin coherence time in order to detect something, because that's assuming essentially that this mechanism is just operating and operating for this time. But nature is very happy to destroy what it has in a measurement and then rebuild it for the next measurement, which we don't want to do when we build sensors. So our mindset is very much in like, how can I build something that stays? But nature may just be happy to destroy the measurement over and over and do it often enough so that you can get something out of there. That's a bit like Daniel Kapnick's idea. And so I think this is almost like a paradigm or dogma that you need to have that spin correlation time. There may be ways to work around this, and I think that's what we need to figure out at the moment.
Yeah, that's what I was talking about when I was talking about this repeated activation through specific thermal conduits, which is generating what you need, and then it decays rapidly, these femtosecond decays, and you keep on doing it until you get the readout that you want on a biological time scale, which is more like milliseconds. So, yeah.
Thorsten, there is a question from Young Chun Kim, who has just joined the Surrey Center for Quantum Biology as a lecturer. And his question is, could you comment on the possibility whether the magnetic receptor might play a role in terms of navigation in ocean creatures?
Oh, yeah. So, I mean, okay. So we have these competing ideas of there being iron oxides, and there's some bacteria where we know that, you know, they form iron oxide crystals to find up and down actually and find an oxygen gradient. There are some measurements from around like 20 years ago in fish with magnetic force measurements that seem to suggest that they do have, you know, a system based on that. For a long time, people have said that if you have a compass that is light independent and that actually detects the polarity of the magnetic field, that may likely be based on magnetite, and fish may conform more to that idea. Birds, on the other hand, have a light dependent compass that also, if you switch the polarity, doesn't change the direction that you're going, and that matches the symmetries of the radical pair mechanism better. The exception are sea turtles that are both light dependent and have a polarity compass. So I think my viewpoint is that it's entirely possible that there are multiple receptors and different mechanisms, and it depends on what you want to do. I think if you only want to detect a 50 microtesla field, there may be, different forms where you can evolve things and make that happen. But if you want to go down to 10 or 100 nanoteslas, I think we're much more challenged. And so I mentioned the spiny lobsters and their homing behavior. So whatever gives them that information, I would suggest, cryptochrome could very well be, or whatever that 10 nanotesla thing is, could very well be involved in this. So my working assumption is that when we find the 10 nanotesla receptor, it's going to be one that is everywhere. And then what it's used for, whether that's useful for animals, that will then, the ecology, I think, will decide whether that's in a given animal or not. But it's the useful thing to have.
Well, I think it's time for us to close this very nice invited symposium on our online version of the APS March meeting for 2020. Thank you all the presenters, and we will be uploading the talks, the slides, and I will post on Twitter, and I will publicize also within APS the places where you can find all of those materials and this nice discussion. And I hope that was a nice opportunity for us to start discussing quantum biology among physicists. So thank you very much. We had 34 people, up to 34 people in this room. So thank you all for participating. And I hope to be interacting with many, many more of you in the near future. Thank you all.
Thanks very much, Clarice.